Good afternoon, everyone. Honorable Vice Chancellor Dr. Shashank Dalvi, Pro Vice Chancellor Dr. Nitin Kadam, Registrar Dr. Rajesh Goyal, Dean MGM Medical College Dr. G.S. Narshetty, and our Director MGM School of Biomedical Sciences. I, Dr. Mini Molpi, Coordinator, take this opportunity to welcome you all to the renowned speakers and participants joining across India and abroad as well, to our national webinar on An Insight into Art Basics of Embryology, organized by Department of Clinical Embryology, MGM School of Biomedical Sciences, MGM IHS Navi Mumbai, to explore the areas of infertility and art in the field of reproductive medicine. Now I request our Honorable Vice Chancellor, Dr. Dalvi sir, to grace the occasion by giving few words.
Good afternoon, everybody. The Director of MGM School of Biomedical Sciences, Dr. Mansi Thakur, the Professor in charge of Department of Clinical Embryology, Dr. Minimul, all other faculty members of Biomedical Sciences, and the guest speakers on this occasion who are going to guide in this particular webinar, and all the participants who have joined for this particular webinar. It is very, very important topic, and I am happy that our biomedical sciences department and these particular clinical embryology are very vibrant units of our organizations, and they are working very hard in the COVID pandemic, and in this also the continuing education for this on this particular important issue. They have organized this particular webinar, the basics of embryology, and the eminent speakers which are joining for this particular webinar, Dr. Kirsi Awari, Dr. Pratiksha Joshi, Dr. Miss Doyle Bose Pandey. All these will be guiding you in this particular webinar, and I am sure that all the participants will surely get the benefit from this particularly joining of the webinar, and I am happy that I hope on this platform you can also interact with the faculty members also, and you can solve your queries also. So I wish all the success for this particular webinar, and I will not much come into this academic feast that you are going to enjoy in this afternoon. All the best for the webinar. Thank you very much. Thank you for inviting me. Thank you, madam.
Thank you very much, sir. Now, I welcome our first speaker, Ms. Doyle Bose Pandey from Indore. Madam is a course director and trainer at Embryology, an academy for learning hands-on embryology at Indore. Her chief areas of interest are oocyte vitrification, ICSI, embryo pooling for poor responders, vitrification, and warming of gametes. Currently, Madam is working at Indore Infertility Clinic as embryologist. She is also a clinic manager, managing all operations pertaining to an independent IVF clinic setup. Her love for learning new things made her master embryology in a short while. She has trained many embryologists over the past and believes in making IVF treatment at affordable for all. We welcome you, ma'am, and her topic for the session is on embryo culture environment, mimicking nature in IVF lab. We welcome you, ma'am. Kindly take over the session. Any queries or questions can be posted in the chat box. Kindly all participants to ensure that audio is muted.
First of all, a very warm welcome to all of the delegates, and I'm very happy to see such a participation just for academic interest. And thank you, Dr. Mini, for such a kind introduction. I feel very important with your introduction. So my experience in embryology has been nearly 6 years, which is minuscule when you compare with most of my counterparts. However, my learning experience has been very recent, so I exactly know how anybody who's new to embryology feels when how overwhelming it could be when you are studying embryology and trying to work in an IVF lab. So I hope that with this topic, it is very much a fundamental topic of embryology, and I hope I have tried to gather all the information in a presentation. And with this presentation, I hope most of you have more clarity than confusion. And if you have any confusion, through the Q&A session, hopefully I'll be able to solve that. So without wasting time, let's get going with the presentation and let's see what we all will be covering today. So even before we try to understand what we need to do in an IVF lab in terms of an embryo culture, we have to first understand what happens within our body, what happens in vivo, and what is the journey that an oocyte or an ovum, right from the time that it has been released till the time it is fertilized, and when it passes through the oviduct and it reaches the uterus for implantation, to know this entire journey and what are the different environmental factors that the embryo faces during this journey. And we will try to mimic the exact same thing within our IVF lab, and hopefully if we are close to what happens in vivo, we should be successful with good quality blastocyst, which will eventually lead to a pregnancy and a live birth. Now, the second thing that we need to understand is what are the different components in an embryo culture system. The culture medium, which is again, if not everything of embryo culture system, it is definitely a central part of the embryo culture system. To understand what are the different embryo culture protocols and strategies which are available, and some basics about culture condition, and some take-home message. Now, with this, okay, I think somebody needs to mute their mic. Okay. Now let's see what happens, what kind of environment an embryo faces in vivo. So in vivo, the in vitro culture simply tries to mimic this environment. And firstly, we see that we have balanced nutrients. So there are a few nanoliter of oviductal fluid which is available to a fertilized oocyte from the time it is being fertilized till the time it is implanted. And this balanced nutrient is being tried to replace that with the embryo culture formulation. Then we have an optimum temperature, humidity, pH, and osmolality that the oviduct provides to this initial compaction and post-compaction embryo. And all of this we try to achieve through a culture media, a buffering system for maintaining pH, and a robust incubator to give you that kind of a humidity, temperature, and concentration of different gases. The third thing which is important in our in vivo system is the optimum amount of oxygen and CO2 tension. And this we try to achieve through incubators. Have, our body prevents from external contaminants and oxidative stress. And we try to constant motion helps the embryo. And this we try to achieve through embryo culture protocols, culture strategy, and an embryo culture platform. There are different kind of suffixes which have been used to the word culture, like platform, system, protocol. I hope with passing slides, we are able to concentrate on one thing and understand what a platform means, what embryo culture system means, and all. And if there is any doubt, we'll definitely solve at the end. Now, trying to elaborate the journey of an oocyte from the time it is being fertilized till the time it reaches our uterine wall, as we see, we can divide this journey into two parts: a pre-compaction embryo, which is day 0 kind of, and a post-compaction embryo. After day 4 till the time it reaches the uterus and implants somewhere around day 5-6. So there are stark differences between the environment, the immediate environment of the embryo, the pre-compaction and a post-compaction. So pre-compaction, the embryos are in control of maternal genome, and post-compaction, the activation of embryonic genome occurs. Now, what is maternal genome and what is embryonic genome? So these are like buzzwords, right? So I'm not going to get into details of genomics because that is a very detailed and thorough topic. But I think to just put in simplified analogy, just like a mother handholds her children when they are born to teach them how to eat, drink, clean, and so on. And then at the right moment when she feels that the child will be able to do these things on their own, she just leaves them to do this under her supervision and then finally eventually leaves it completely. So maternal genome is kind of pretty much the same thing. Here, um, the gene products of the maternal, the mRNA and all, they regulate the initial meiosis, fertilization, and the early development of the embryo. It is kind of hypothesized that it is the maternal genome which indicates and the switching over from the utilization of the maternal RNA to the embryonic genome occurs, and this is triggered, this is controlled by the maternal genome itself. So this is called the zygotic genome activation. Even if you don't understand too much of the details of maternal and activation of embryonic genome, I think what is most important to understand here is pre-compaction embryos are very susceptible to their immediate environment. So they are very susceptible to minor changes in pH, temperature, and osmolality. While as the post-compaction embryos are much more robust because of the number of cells that they have, and they can regulate temperature, pH, and osmolality much better than the pre-compaction embryos. And hence, pre-compaction stage, the best is them untouched as far as possible. So don't just fiddle around things. Then be very gentle with your gametes and leave them as much untouched as possible. Other major difference is the energy source or the energy substrate. The initial energy source during the pre-compaction stage, the main source of energy is pyruvate and lactate. It's not glucose. While as in post-compaction stage, the source of energy or the energy substrate is. That's why the metabolism initially is called an oxidative metabolism, which is more quiescent, and the metabolism in the post-compaction stage is a glycolic metabolism. The third difference, which is a major difference, is how non-essential amino acids are used in the pre-compaction stage, and both essential as well as non-essential amino acids are needed in the post-compaction stage. So now you guys know probably amino acids better than me, but there are around 20 or 21 amino acids, and non-essential amino acids are the one which your body cannot synthesize. So sorry, essential amino acids are the one which are the ones that you cannot synthesize on your own. So the cells initially will not want to be dependent on something that you cannot synthesize. So initially, you will want to be dependent on things which are available to you readily, right? So this is the major difference between a pre-compaction and post-compaction. And also you have differences in the temperature, pH, and oxygen tension. Like shown in the slide, the temperature in the pre-compaction stage is about 35.5 to 36 degree, while in the post-compaction stage, the temperature which the embryos are subjected to are close to 37.2. So there is this kind of 1 degree variation in the temperature between the pre-compaction and the post-compaction stage. So at the site of fertilization, it's important to note that the temperature is slightly lower, which is 35.5 degree. The pH also is different. The pH in the pre-compaction stage is 7.94, while that of the post-compaction stage is 7.2 to 7.3. While as we go forward in the slides, you will see that the pH is actually a high, low, high kind of a paradigm, which we'll see later on. And the oxygen tension in the pre-compaction stage is approximately 5%, while that of a post-compaction stage is approximately 2%. So let's move further. Now, embryo culture system. So everything which comes in close vicinity to our embryo is a part of the embryo culture system, so including us, the embryologists. So the central part of the, what do you understand by a culture system? There are various factors which come together to give an end result, and even slight or minor variations on one part is going to affect your development of the embryo. So these are the 8. I try to list a few things which are part of the very important part of the embryo culture system. Embryo culture media, naturally the centermost part. Then you have the kind of embryo culture dish that you use, the type and the quantity of oil that you overlay on your dishes, gas phase for equilibration of media, temperature and humidity, filters that you use, air quality in your IVF lab, the embryologist himself or herself, and the type of gamete handling, how precise you are at your gamete handling. These are all part of your embryo culture system. Now, let's go quickly to embryo culture media. Now, today we'll try to cover what are the expectations from our embryo culture media. We know that the first and foremost expectation is that it should ensure growth of an embryo, to decrease the embryonic stress, and it should also maintain the gamete viability. These are the 3 foremost expectations that we have from our culture media. About the history of embryo culture media. If we, instead of going through each of this, I think it is important to note that we started as early as 1929, where first we tried to culture rabbit embryos in plasma. Then slowly and gradually over the next 50 years, we reached step 2, and Edward reached a point where they used Earl simple solution with pyruvate supplemented by patient serum. And this is in the environment in which the first successful test tube baby was cultured. Beyond 1978, a lot of things happened with the embryo culture medium, where Menezes and his friends tried to add a few serum albumin, and that the major thing that they noted was serum albumin resulted in non-adherence of the embryos or the gametes to the glass surface. 1990 onwards, we came to, after the advent of computers, the scientists took help of computer and something called a simplex algorithm for optimization, and they tried to identify and alter the concentration of components which at a higher concentration were detrimental to the embryos. And this gave rise to the called simplex optimized medium, or SOM. And this was the stepping, this is what gave rise to the single step media commercially. And after 1990, we see that 2 lineages of the culture medium. One was a sequential media group, which is called a back to nature, and the other was a single media group, or the one which followed through the SOM culture media. And this was the single step media, let the embryo choose. So we still have, both of these culture strategies, protocols are very different, and we still have a lot of debate on it. People who vouch for sequential, they prefer to go sequential way, and there are many people who prefer the simplified single step media. And both of them have, benefits and disadvantages of their own. So it is up to us how to choose based on some informed decisions. Now, instead of going through the composition of the culture media completely in detail, we won't be going through what are the purpose of amino acids and fatty acids and all, because we will be short of time. We have a lot of things to cover today. What is important to know that commercially, I think there are about 10 to 12 companies who formulate media, culture media. And this new generation culture media has more than 80 plus components in them. These components are majorly divided into these: water, basic salt, carbohydrates, amino acids, fatty acids, EDTA, vitamins and antioxidants, growth factors, antibiotics, etc. and etc. None of these companies, the commercial companies, will let you know the percentage, the exact constituents of the media. That's always a trademark. So but they will tell you that they have, what is the salt that they have used, but maybe not the exact quantity of the salt. Key components of a modern medium. So if we, like we saw that we have one, one media system is the single step, then sequential media has two cleaving media and the blastocyst media. So if we compare a single step media with a cleaving and a blastocyst media, we see that the energy substrate is kind of all of them have glucose, lactate, and pyruvate. However, the quantity of glucose, lactate, and pyruvate would be different for one media, would be different for a sequential and a blastocyst, cleavage and a blastocyst media. The cleavage media should have lesser amount of glucose as compared to the blastocyst media. When we talk about the non-essential and essential amino acids, we see that the one system has all the 8 non-essential amino acids, while the sequential cleavage media has 9 amino acids and so on. This is just a chart mentioning you a few things. One thing which is important to notice, chelators. Chelators like EDTA. So like we see, EDTA is said to have a detrimental effect over plastulation, and that is why in the sequential media, you have no EDTA, while you have EDTA with some, minimized form in the single-step media. Vitamins, we have, again, this is one chart where it is showed that the sequential blastocyst media has 4 vitamins in it, while it is not present in the single step and the cleavage media. The purpose of vitamins, again, it has a lot of antioxidative properties. So this is just a charting. We don't need to learn this by heart. This is just, something which you should do in culture media when it comes, when you open the bottle. This is a kind of chart that they will mention. You can, usually we don't even look into it. But the constituents of the chart will be something like shown here. Now let's go to culture media type and protocol. So we were talking about back to nature or sequential culture and let the embryo choose or a single step culture. These are the 2 basic type of culture protocols available, and we'll also see a comparison between the 2. So when we say back to nature, we want to mimic the in vivo environment of the oviduct as much as possible. Because we already saw that pre and post compaction embryos are subjected to a very different kind of an environment. We also saw that how the maternal genome is active and later on the embryonic genome takes over. So, and we also saw pyruvate and glucose preferences. We saw that the embryos initially not use glucose, they use pyruvate and lactate, and in fact, glucose in the initial culture environment, when we tried to do it in our, from 1920s and on, glucose used to have this 2-cell block if you added too much of glucose in your culture media. Then we have role of non-essential amino acids and essential amino acids in the pre-implantation embryos, and inhibitory effect of EDTA on blastulation. So people who developed the sequential culture media saw all of these and tried to implement 2 different kinds of media. They said that the energy requirement is changing in the pre and post, and glucose in all species does inhibit the 2-cell block. So changing the quantity of glucose in the cleavage and blastocyst media is necessary. They said you cannot have one media which will have glucose at a similar level, since your embryos need a different type of energy substrate initially, have a cleavage media, pre-compaction, and that should be changed or renewed post-compaction by the blastocyst media. They also took care of EDTA, saying that has a detrimental effect on the blastocyst development. So they removed EDTA altogether from the blastulation media. they hypothesized the role of amino acid on pre-implantation embryo, that the cleavage media will contain only non-essential amino acids because the essential amino acids are not needed at that stage, whereas the blastocyst media contains both NEAA. Another important factor which was seen was chemical breakdown of L-glutamine, which is a kind of an amino acid again, that in aqueous solution, when L-glutamine would break down, it would accumulate a lot of ammonia. And this ammonia has detrimental effect on the embryonic growth. So they asked to renew the media, and so that the accumulated ammonia could be gotten rid of. So in sequential culture, as a rule, you renew the media. Sometimes some people do it, on daily basis, but not anymore. But you usually renew it on day, and that way you get rid of the accumulated ammonia. Now, this is in this debate, single step and monostep culture, which was based primarily on the works of Driggers and Cooper. They said that let the embryo choose. As long as concentrations of all the things like glucose, amino acids, EDT, as long as all these concentrations are within tolerable ranges, the embryo itself will adapt to its environment and utilize whatever is needed. And their strongest point that they argued on was the pathophysiological stress. When you switch an embryo from one kind of a media to the other kind of a media on day 1 and day 3, you, you know, it has a lot of stress. There are osmotic and pH changes, temperature changes. All of this could are stress factors for the embryo, and will be depriving the embryo of some autocrine paracrine factors that may have been produced during the first culture period. They also had said that glucose is present in the natural environment in the fallopian tube itself, and there is evidence of culture media with glucose that do not inhibit embryo development. So, say experiments they cultured embryos with glucose, and they found that it does not have any detrimental effect. The role of EDTA. The negative effect of EDTA during blastulation refers to a higher concentration, but the presence of low quantity is not deleterious to the culture of the embryo. Use of mixture with all 21 amino acids. This way, the embryo would be able to choose which one to use by itself. So instead of first giving one kind of amino acid and then the other kind on the cleavage and blastulation media, the single step media provides all the 21 amino acids and lets the embryo choose whichever amino acid it needs. The problem of accumulated ammonium from decomposition of GLN is resolved by using a more stable dipeptide of GLN. These are called glycol L-glutamine or L-alanine L-glutamine. So the accumulation of ammonia was said because of the breakdown of L-glutamine. So if you replace it with a more stable dipeptide, then there will be no accumulation, and single-step media should work. You should not have to renew it just because of the concern of ammonium accumulation. Now, a brief comparison between culture period of commercially. I'm sorry. Uh, Sorry for that interruption. So, comparison between the culture period of commercially available media. You have a one media system and sequential media system as mentioned here. So these are a few companies that provide you single step media and a few companies that provide you sequential media. What is important to note here is the culture period. The culture period of this is something very important that you should know the moment you get an embryo culture. Any new media that you get, you should know from which day you can culture your embryos in it. So like you see, some of them say day 1, some say day 0. So this is the most important part here. that says that you can culture it from day one. You cannot, just fertilize an oocyte and leave it in. It is a cleaving media where you have to transfer it on day one post fertilization, post the 2 p.m. check. While mediums that mention day zero are the ones where you can simply, do an ICSI or post a long incubation, you can simply keep your embryos in there for extended culture. Now, day zero, day one, like I mentioned, it is important to note that many media do not recommend culturing oocytes in them. So if you keep your oocytes in them, then it will be detrimental for the oocytes and they will not perform, it could affect its fertilization later on. Now, sage IVM, if you call about in vitro maturation of oocyte, then sage IVM medium is um, the only kind of commercially available medium which is meant specifically for in vitro maturation of oocytes. However, studies have shown that you can successfully use blastulation media for the same. So at times, if you get all immature oocytes and you want to attempt uh, you denutate those sites and they are all immature. The only choice that you have left with is probably do an in vitro maturation. At that point, you should use a sage IBM medium or a blastocyst medium. Not be the blastocyst medium may not be as good as the say which is specifically designed for IBM, but it is a good choice in absence of an alternative. These are the kind of protocols that we usually follow in our lab. So in a sequential media, you have an uninterrupted culture till day 3. And after day 3, move it to the blastulation media, if at all you are going for a blastocyst culture. Now, sequential media with a fresh state-specific medium, this no one follows it. I haven't seen any lab that follow it. There are single step, then the other one is the single step or a mono-step culture. And the same media is used throughout the 5-6 days. And single step media with an interrupted culture where you do a media renewal on day 3. So there are many labs who go for a single step media because of the ease of protocol. And yet they change the media on day 3. This is their own protocol. Maybe they have seen some differences in blastulation and all. That's why. Now, if you have to compare the protocol, so based on these characteristics, we are trying to compare a single step or a mono step culture protocol with a single step interrupted or renewed protocol and a sequential with renewed. You see a lot of smiley points and a lot of sad faces. Now, the main factors, which, these are the factors based on which any IVF lab should decide what kind of protocol they want to follow. So, leaving the embryo undisturbed, everybody should ideally do it, right? But, you see that at the cost, it is at a cost of higher blastocyst, lower blastocyst rate and all. Maybe you would decide to go for a sequential media. This protocol varies lab to lab. Replacement of essential nutrients, accumulation of toxins, environmental stresses. I think this is a very important factor. The environmental or the pathophysiological changes because of the actual physical move of the embryo. Then labor intensity of the embryologist, which most of us are very concerned about. Single step uninterrupted culture is the easiest thing to do. So if you have new embryologists, if you have embryologists who are trainee and not that proficient, I think you want to choose this protocol. Again, cost, time management, and QA/QC. All of these factors needs to be considered before you actually decide on a protocol. Single step media are less expensive because you don't need 2 kinds of media for a single patient. You just need 1 type of media. So these are the factors that you have to weigh in and then decide which protocol you want to choose. So after going through all this, which protocol is superior? basically the studies are completely inconclusive. So if you are doing sequential and it is working for you, you should stick to it. And if you are doing single step and it is giving you an industry, benchmark standard of fertilization and blastulation, then you have no reason to worry or thinking of. After the embryo culture protocol, we come to the embryo culture strategies. Embryo culture strategy is whether you want to culture individual embryo individually or you want to culture them in a group. What the various, and we want to see various advances that has happened in the culture strategies. These are the kind of dishes that every IVF lab uses to culture their embryos. So you have single embryo culture, for which you will make droplets and then you will keep embryos in single droplets, or you have specialized dish these days where instead of forming droplets, they have micro wells in which you can keep your individual embryos. And okay, let's briefly see what an individual culture, what are the advantages and disadvantages before we actually choose one type of a strategy. Advantage for an individual evaluation, the first and foremost advantage when you do an individual culture is you can individually evaluate an embryo morphology and its rate of development. This is very important. If you do things in a group, you won't be able to tell. You won't be able to tell or mark the morphological characteristics over the course of 3 to 5 days of development. You won't be able to do that. So this is the most important advantage of an individual culture. Now, embryos that are cultured together may be exposed to secreted detrimental factors too. So if you have one embryo which is apoptotic, some cells, then if you culture them in a group, then the detrimental effect of the embryo which is is going to apoptosis might affect the embryos which are good grade. And depletion of necessary substrates by other embryos if cultured in a group. So whatever is a negative part of a group culture is advantage. It comes as an advantage of an individual culture. So we say that when you culture them in a group, you can also, the embryos could end up depleting the necessary substrates which are present in the culture medium. Talking about the disadvantage, the individual culture is again labor and cost intensive. Special kind of plating at times, if you happen to choose that, adds to the cost. And the 15, 16 sites and trying to keep them in different micro droplet could be labor intensive. However, with practice, even practice doing this, then it doesn't seem cumbersome. And loss of autocrine, paracrine growth factors affecting growth. So, embryos like being together, they don't like being alone. So when there are things called autocrine, paracrine factors and growth factors, which every embryo, they share among themselves and it enhances their own growth. So if you culture some embryos individually, you might lose on the autocrine, paracrine and growth factors. Temperature and osmolality during the manipulation of this could be a concern because individual droplets have few microliters of embryo culture media. So smaller the droplet or smaller the quantity of media, more difficult it is to maintain its temperature, its osmolality, as well as its pH. If you have a larger amount of the media, it's much easier. So what are the advantages of group culture or communal culture? Let me see, there I am on the timeline. Okay. It's an easy protocol. I'll have to be fast, I think. Okay. Group culture or communal culture is easy protocol. You use accumulated autocrine, paracrine growth factors, and you have less exposure to temperature changes during the manipulation. Disadvantage, it does not permit continuous evaluation. This is the biggest disadvantage of group culture. You cannot individually evaluate them using time lapse or or even individual marking individual morphological characteristic is not possible. And the embryos in a group may be exposed to negative effects of a dying or a delayed embryo, which are cultured in the same well. So benefits of group culture are more evident when you plan a blastocyst transfer. So this is important to note. A group culture benefit usually comes into picture only when you are planning a blastocyst transfer or an extended culture. Now, these are some advances in the embryo culture platform. I think I'm just mentioning that you have things like rotation, shaking, pivoting, vibration, and piezoelectric actuators. We are not going into detail of these are few advances which is the future of the culture platforms which are being looked into. So let's quickly move to temperature, pH, CO2. There are culture conditions. What can so most important temperature, pH, CO2, and oxygen tension. So, like we had seen in our slide of a pre-compaction and post-compaction embryos, there's a difference between the temperature even within the oviduct at the site of fertilization to the time where it is implanting in the uterus. So, what is important to note here is the minimum temperature may not be as critical as the maximum temperature. There is something called the quiet embryo hypothesis, which says that viable embryos have a quiet metabolism, and cooler temperature, they say, could lead to a quiet metabolism. This is just a hypothesis, by the way, because the randomized control trials are inconclusive. But again, having said that, and even noting the fact that fertilization site is 1 or 2 degrees less as the core body temperature, it is important to note that the minimum temperature may not be as critical as the maximum temperature that we are subjecting our embryos to. Reduction in temperature will only slow down the enzymatic function. But rising temperature, even by say half a degree or degree, what's the effect? Your oocytes maturation, the spindle formation, the spindle might not form back, and it might affect your fertilization rate and retards further embryonic development. Coming to the pH. So pH is study of pH is just like chemistry. It's actually complete your undergraduate level chemistry. pH in the media. Now again, pH is differential within our oviduct. The site of fertilization has a higher pH, site of cleavage has lower pH, and blastulation is again higher pH. So we have a high, low, high pH. Now, acceptable pH range that we culture our embryos into is usually set from 7.2 to 7.4. These are the various factors which affect the of an embryo culture media. It is the amount of bicarbonate which is present in the media, the concentration of amino acids which are added to the media. Now, the first 2 points, the amount of bicarbonate and the concentration of amino acid, we can't do anything about it. This is the formulation of the culture media. The culture media is delivered to us. We are not making it, right? So what are the other things that we do that impacts the pH? So how much volume of media are we, sorry, how much volume of the media we are taking? What is the oil overlay? What kind of oil are we using? How much oil are we using? Kind of culture dish that we are using, the altitude of the lab, the CO2 concentration in the incubator, and temperature. This all affects your pH. And like we see, there's an inverse relationship between pH and temperature. So when you rise your temperature, the pH lowers, and when you rise your CO2, the pH again lowers. Now, how do you change pH without actually changing temperature and CO2? Already saw we have incubators where you maintain a particular temperature, you set that value, and you maintain a particular percentage of CO2. Don't change it on a daily basis. Now, having said that, what happens? How do you change pH without changing the temperature? Because we have to be close to the high, low, high of, like we saw fertilization, cleavage, and blastulation. We do this by changing the bicarbonate concentration in culture media, and this is what primarily the sequential culture media group emphasize on. So they have a different bicarbonate concentration in their cleavage media, while they have a different one in their blastulation media. And even keeping the same this media in the same incubator with the same set point of a CO2 and temperature, you can achieve different pH for the culture. Now, like how pH measurement of pH ideally should be done every day, but it is very cumbersome. It is easier said than done. The instruments for measuring pH are expensive. So I'm pretty sure that most of the labs, yeah, and even outside India, even in Europe, they don't regularly or every day measure the pH every day. The easiest way of dealing with whether the pH is right or wrong is by giving it a visual indicator. Some culture media, not all though, some culture media give it a phenol red indicator. A phenol red has a property of changing its color depending on the amount of pH. At very low pH, phenol red is almost colorless, while at a higher pH, it has a bright fuchsia color, like the one which is shown in this culture media bottle. How do you regulate pH? A buffer is something that is added to keep the pH of a solution constant. These are the two definitions that you need to just briefly understand. Buffering capacity is the resistance to changes in pH when a strong acid or base are added. So HIPIS and MOPS, these are two buffers. So we saw that, in our previous slide, we saw that you can maintain pH. If you have a culture dish with culture media, you keep it in the incubator, you maintain a temperature and you maintain the CO2 concentration. It will be stable, right? Now, this is fine as long as you're just culturing it without having to take it out. Now, there are certain things like, denudation of an oocyte and even screening your OPCs for that matter, certain kind of gamete handling that you have to do outside the incubator. How do you maintain your pH when the gamete is outside the incubator? So this is where buffers come into picture. So you have this buffer media in which you do all the gamete handling. Now buffer media are the two most successful buffer medias are HIPIS buffered media or HIPIS does seem to have buffering capacity which is closer to our physiological pH that we need. However, what is important to note here is the moment you take a dish outside an incubator, the temperature is going to fall. It's not going to be 37 degrees. And since pH and temperature have an inverse relationship, the moment the pH falls, the buffering capacity, which is 6.93 of MOPS, the pH is going to rise and probably it's going to be somewhere 7.2, between 7.2 and 7.3 that we are targeting. So HIPIS and MOPS buffered medium are also Zwitter ionic buffer or good buffer. They are named after the scientist Norman Good. And at times we also, these days we have also seen that instead of using just simply HIPIS and MOPS, you have a combined buffer, which is a mixture of HIPIS and MOPS, and sometimes some other kind of buffer. Like for example, we have a 1:1:1 for HIPIS, MOPS, and DIPSO. Now quickly coming to the oxygen. First, noting down fact. The fact is the embryo passes through sequential oxygen concentration, which varies from 5% to 2%. It is not constant at all. The optimum oxygen tension of the in vivo embryo is dependent on the stage of development, whether it is on a pre-compaction stage or post-compaction. And post-compaction embryos benefit more from lower oxygen concentration. This is also a fact. Now, in vitro embryos are subjected to, this was the fact for what happens in our body. Now if you compare it with what we are subjecting to in the lab, then in vitro embryos are subjected to a monophasic oxygen concentration. Why? Because you're keeping them in an incubator which has a only, it's monophasic. You can give only one particular concentration, either atmospheric or if it's a dry gas, a 5%. Embryo culture at 5% oxygen results in improved morphological parameters, reduced embryo arrest, and higher live birth rate as compared to atmospheric oxygen concentration of 20%. There is abundance of data which shows that atmospheric oxygen negatively affects the pre-implantation stage. So these are the two type of incubators that I have tried to show here. This is a box incubator. It looks like a box. And this is a benchtop incubator. So coming to box incubator typically uses an atmospheric oxygen concentration of 20%. So in the room around us, we have 78% nitrogen, 20% oxygen, and 0.04% CO2, right? So the moment box incubators will use one kind of a gas, CO2 cylinder, to maintain the CO2 level. However, they will rely on the atmospheric level of oxygen. So the moment you open the door of a box incubator, the atmospheric air will rush in, and in order to, and then the CO2 incubator has sensors in it. So it will automatically reduce the CO2 level. So the sensors will detect the reduction in the CO2 level and appropriately will purge the CO2 gas again back to it so that it comes to a 6% level. And this means that the oxygen level will still stay at close to 20%. Now, the box incubator initially, which was used for somatic cells, were larger units of 150 to 200 liter. However, dishware, which is used for IVF, is considerably smaller, and hence even a 14 to 48 liter box incubator should work for the media equilibration and culture. Important thing to notice: if you want to buy a bigger box incubator for culturing more dishes, for example, or equilibrating more, have to remember that it also influences the gas equilibration and the recovery time. So it takes more time to recover. Coming to benchtop incubators, benchtop incubator typically use 5% physiological oxygen. They have extremely small chambers of 0.31 to 0.5 liter, and this improves the recovery time a lot. Smaller incubator volumes are beneficial in terms of rate of atmospheric recovery following repeated opening. The box incubators, and when you compare with box incubator, the recovery time of benchtop incubator is fast. Most benchtop incubators will utilize cylinders of pre-mixed gases. So the tri-gas cylinders, which has a pre-mix of 6% CO2, 5% O2, and 9% nitrogen. A modern triple gas incubator, though, has a cost implication. So it is not only the first time cost which is of consideration, it is also the renewal of these gas cylinders. When compared with the CO2 cylinder, a tri-gas cylinder is much more expensive. Nowadays, like we saw that the blastocyst in a uterine cavity is subjected to only 2% oxygen. Ultra low concentration of oxygen is being studied upon, but evidence is still inconclusive. So this brings me to the end of my presentation and the take-home message. If in doubt, always stay towards the lower temperature. Say you don't have a temperature meter, you have touched a surface or whatever, you feel the temperature is high. Staying a tad bit on the lower side is better, far better than being on the higher side. Best in a benchtop incubator, 5% oxygen concentration is what our body gives to our embryos in vivo. Let us try to match it instead of showing studies which say that it has no effect on blastulation and all that. I think we should all invest in benchtop incubators, and it should not be something that you should purchase only when later on. On the first go itself, purchase a benchtop incubator. Simple things often work best. So simplify your workflow. Don't have a very complicated workflow, because if you have a complicated workflow, it will be very difficult to replicate it for more number of cases. Reduce gambit handling, touch less, as less as possible. Take the dish out of incubator as less number of times as possible. If your cumulative results are good, the word cumulative here is of uttermost importance. One batch giving you 80% result should not have you on the top of the moon, while at the same time, one batch giving you just a 20% result should not you should not be depressed. You should just go back to troubleshoot and find out what might have gone wrong. So results should always be looked upon cumulatively, more so over a year, so that you have covered all seasons. Lastly, do not be afraid of a change, but change only one thing at a time, even if it is something as trivial as a Falcon tube or one kind of a buffer. So just one thing should be changed at a time. Monitor it for a while, and if you are happy with the result, then go and change the other thing. So this is the end of my presentation, and a few references, which was very interesting. If you guys want, I can share this later with you. Ma'am, I think I'm done. I'm ready for question and answers.
Over to Rafia for question answer session. Please, Rafia. Unmute.
Sorry. Good afternoon, all. I'm Rafia Sangameshwari. Ma'am, I'll read out few questions for you.
Yes.
So, to start with, how is oxygen, tension, and pH of media is adjusted in single-step media?
How is oxygen tension adjusted in single-step media?
Yes.
You also said pH?
Yes, ma'am. Oxygen, tension, and pH of media.
Okay, so oxygen tension, it depends where you are equilibrating the media, right? So if you have a single step media and you're equilibrating it in a box incubator, for example, right? So your oxygen tension in a box incubator is 20%. So I think if I'm not wrong, it takes approximately 7 to 8 hours to equilibrate your media in a box incubator for the oxygen tension and pH. And it also depends on the kind of protocol that strategy that you're using. If you're using, say, a 4-well, the equilibration time will be different. While you're using a center well, it will be different. If you're a micro droplet, it's a different case altogether. But safely, you can say if you keep it for at least 6 to 8 hours, then the media will be equilibrated for the oxygen tension and the required requisite pH. Does that answer your question? I don't know whoever asked.
Ma'am, can I ask next question?
Yes, please.
Yeah. Can oxygen and carbon dioxide level be altered for culturing?
Again, depends which incubator you have, right? So if you theoretically, box incubator, you are only providing CO2, right? You have CO2 cylinder and 6% is your target set value for your CO2. And rest of it takes from atmosphere, which is atmospheric oxygen concentration of 20%. So did you, Rafia, can you just mention, was it oxygen and CO2 both? Was the question
Yes,
both?
yes, ma'am. Oxygen and CO2 both.
If you want to change the CO2 concentration for whatever reason, experimental reason you have to, you will just have to set it to, instead of 6%, you will set it to whatever, say 7%, 8%, incubators let you set that. So when you do that, it is going to use appropriate amount of CO2 from the incubator, and that can be available within your incubator. If you talk about a benchtop incubator, it has a pre-mixed gas. These days, you also get benchtop incubators with mixers, where you have to give it nitrogen, CO2 and O2, 3 different things, and it is going to mix it. Actually, you give 2 things and it mixes it. But most of them use trigas, which is a pre-mix of 6% CO2, 5% O2, and rest of it is nitrogen. So if you have to change the oxygen and CO2 concentration on the benchtop incubator, you will have to get a different premix. You have to go to the manufacturer, ask them to give a different premix, saying that I want to try 2% oxygen for example. So you have to go to the manufacturer of those cylinders and ask them to give that to you.
Okay, ma'am.
Any more questions, Rafia?
Yes, ma'am. One more question is there. Does culture media influence the mosaicism?
Mosaicism?
Yes, ma'am.
I would really want to pretend and say I exactly understand genetics that well, but trust me, it's a very huge kind of a thing to say. I think, as far as my knowledge is concerned, there is epigenetics is such a topic which scientists को नहीं समझ में आया, मुझे क्या समझ में आएगा? I think it does have some impact somewhere, but mosaicism is again something some embryos will, more than culture media, I think it is the culture strategy which could, but I'm not very sure on this. I'll have to go find out papers which actually say whether it actually can lead to, some cells being chromosomally normal, mosaicism, while some being, aneuploid, while the some being euploid. In general, an embryo itself, a blastocyst itself, has a lot of mosaics. Not all, you are just, who has taken every cell from a trophectoderm of an embryo and and ran it on a next generation sequencing to see if all of them were euploid or not. None of us have done it. What we have done so far is just taken 8 to 10 cells from a trophectoderm and considered that as the best representation of our embryo and run it through, PGT and seen whether they are euploid or not. And mosaicism, I'm not very sure if a culture media can actually impact it. More so of ICSI or not ICSI and things like that might be more questionable in that.
Okay, ma'am. Ma'am, one more question.
Yeah.
What is exactly mechanism of low oxygen use for embryo culture?
What is the exact what? Sorry.
What is the exact mechanism of low oxygen use for embryo culture?
Mechanism. I think it is more to do with how the embryo, the glycolic metabolism takes over. So, whoever asked this question, I think Rafia, you would know that who asked the question. उसको पूछो कि कितने अच्छे से अपनी theory पढ़ी है. I think it is more to do with the glycolic metabolism, and within that, I am not very proficient with the cell, the chemistry of it. But lower concentration of oxygen will give you it is a kind of aerobic, anaerobic, मुझे अपना revise करना पड़ेगा metabolism which kind of does it, but I'm not very confident on that bit. So you have to go back to your textbook and see the kind of metabolism that a blastocyst, the smaller cells, do the uptake of glucose and how they form about their cells and all that. I'm not very proficient with that.
Okay, ma'am. Thank you so much, ma'am.
Welcome.
Over to you, Mini ma'am.
Thank you, Rafia. Thank you, madam, for such an elaborate talk on embryo culture. Indeed, it was very informative. Thank you, ma'am.
Welcome.
I now welcome our next speaker, Dr. Pratiksha Joshi, Director and Chairperson, Senior Clinical Embryologist at Ma Women's Hospital and IVF Center, Ahmedabad. Madam is a doctorate in clinical embryology and microbiology, PG in clinical research and PG in health and hospital administration. She has also done her fellowship in cosmetology, presented over 50 lectures in national and international conferences, worked as a scientific officer at IKDRC IVF department for 11 years. She's responsible for more than 2,000 ICSI babies. I welcome you, ma'am, to take over a session on ICSI, the ultimate to combat infertility, the male factor.
Thank you. Am I audible, madam?
Yes, madam, you're
Okay, yeah. Thank you so much, madam, for such a nice introduction. First of all, I would like to thank Dr. Sasank Dalvi sir, Dr. Nitin sir, and Dr. Mansi madam, and especially Dr. Mini Mall madam, and all the respected speakers and delegates. I tried my level best to prepare this presentation as simple as possible. Intracytoplasmic sperm injection, ICSI, the ultimate combat infertility, especially in male factor infertility. So, as all we know that nowadays, infertility is increasing day by day. And what is the definition of basic infertility? That is unable to conceive after a 1 year of unprotected intercourse. And nowadays in India, it is higher, like 40%. So, if we talk about the male factor infertility, according to the scientific data, it's divided in infertility causes 30% from the male factor, 30% from the female factor, and 25% there might be unexplained infertility, 10% combined factor, combined both male as well as the female, and 5% other factors. So the major causes of male infertility include some semen disorder, morphological disorders, testicular failure, obstructions, varicocele, undescended testes, and some erectile dysfunction, and the unknown factors. So why this infertility is increasing day by day, both in male and the female? The reason being delayed marriage and late childbearing. Some sexually transmitted disease like HIV, HPSAG, VDRL, etc. Heavy pollution, diet like junk food, canned food, lack of the exercise, and the previous contraceptive use leading to sterility in the female, and sterility from previous abortion. And in male factor infertility, basically falling sperm count in males because of stress, because of alcohol, cigarette smoking, gutka chawing, etc. So here in this picture, you can see that excessive use of electronic devices. Here you can see a heat from the laptops and the radiation from the mobile phone also lead the infertility or the lower down the sperm count in male. Excessive use of packed the drinking water, radiation from the mobile tower, and the use of the plastic also increase the infertility. In male,
Ma'am,
hello,
hello, ma'am. Can
Haji,
you
ma'am.
kindly
Hello.
share your PPT, ma'am?
Yes, ma'am, it is there.
No,
Hello.
we are now not able to see, ma'am.
Just hold on, ma'am. Just hold on, ma'am.
Yes.
એ લોકોને PPT નથી દેખાઈ.
Yes, ma'am.
Now it's okay.
Yes, ma'am. We can hear. Yeah.
Yes.
Carry on, ma'am.
Yes, madam. So, intracytoplasmic sperm injection and ICSI, basically a male factor infertility. As I have already discussed that infertility is increasing day by day, and there are a lot many factors. A couple unable to conceive after a 1 year of marriage life, unprotected intercourse. So in India, this scenario is more increasing up to the 40%. So there are a different type of infertility, both that contribute male 30%, 30% from the female, combined 10%, and some unexplained infertility, like the both reports from both male and female are normal, but still they are unable to conceive, and other factors 5%. So if we talk about the major causes of male infertility, semen disorders, testicular failure, obstruction, azoospermia, undescended testes, and varicocele, ejectile or erectile dysfunction. Those all are major causes of male factor infertility. So, what are the reasons why infertility is increasing day by day? That is because of delayed marriage, late childbearing, some sexually transmitted diseases like HIV, HBsAg, heavy pollution, diet like junk food, canned food, lack of the exercise, previous contraceptive use leading the sterility, sterility from previous abortions also in the female, and falling sperm count in the male, specifically by some medicines, drugs, alcohol, cigarette smoking, gutka chawing, overstress, and excessive anxiety in male factor infertility. Here in this particular picture, you can see the Radiations from the mobile and the heat from the laptop also leads infertility in a male partner. Overuse of packed food, packed drinking water, radiation from the mobile tower, and excessive use of plastic canned food can increase the infertility into the male. Male, that I work in the battery industries, lead, cement factory, also come into the contact with high environmental pollutant and increase the ROS factor into the cement. That can also slower down the motility and other genetic abnormalities and DNA damage into the sperm. As all we know about that, IVF has been started in 1978. We all remember the Louis Brown. We all remember Robert Edward and Practic Steptoe, the two main pioneer. And after that, all new type of technique, ICSI, IMSI, vitrification, laser-assisted hatching, and lot many research introduced into the field of embryology. But the most important thing, our India's first IVF baby was born in 3rd October 1978, and we must not forget the great contribution of Dr. Subhas Mukhopadhyay. But unfortunately, at that time, government did not recognize their contribution, and he ended his life in 1981. But after a few years later, his colleague presented all his research papers and to the government, and after that in 2002 ICMR appointed a 12 members committee research on this subject, and finally in 2003 Dr. Subhas Mukhopadhyay's work was finally recognized and get a official stamp. So what is the primary goal of any type of infertility treatment? That is, give the infertile couple with live and healthy baby. Intracytoplasmic sperm injection, ICSI, is a specific technique, micromanipulation technique, used in process of fertilization. So here in this particular technique, injecting a single morphologically normal live and motile sperm into the center of mature oocyte, ooplasm, with the help of specific types of micromanipulator. So here in this particular technique, injecting a single sperm into the egg cell so that the sperm does not need to penetrate the cell automatically in order to fertilize the eggs. So ICSI is quite different from simple in vitro fertilization. Here, embryologist has to select a single normal motile sperm, and it's directly injected with the help of needle into the eggs instead of fertilization in case of IVF taking place while in a center well dish. Sperm are placed near the oocytes and allowed to fertilization take place into the center well dish. So here in IVF, oocyte select the specific sperm, while in ICSI, embryology select the normal and motile sperm. So which kind of couple need a ICSI? So ICSI basically benefits the couple with infertility due to the severe male factor issue like poor sperm motility, less than 10% mobile sperm in ejaculate, abnormal morphology like head abnormality, midpiece abnormality, tail abnormality, double head sperm or band tail sperm, severe oligospermia, very low sperm count, and teratospermia. Azoospermia, that means a zero sperm count, either due to the vasectomy or other obstruction. Because of the problem with sperm production, sperm have been, because of this azoospermia, no sperm inside the semen. So the sperm can be retrieved in this particular cases by testicular sperm aspiration. And most important factor is problem with the antibodies against the sperm testicular cancer also, and a previous failed fertilization in a simple IVF cycle. So in all those cases, a couple require a higher technique like a ICSI. So if a male partner has a problem in erectile dysfunction or the ejaculatory dysfunction, so when ICSI is used when a small number of eggs are retrieved, when the patient has female has low AMH anti-Murrelin hormone level less than 2, and only 3 or 4 oocytes are retrieved. So in that particular case, patient as well as the embryologist insist that ICSI give a better fertilization rate. And when you are using a frozen semen sample where the quality of the spermatozoa is not up to the mark, in that particular case is ICSI is beneficial for that particular couple. And when infertility is results from poor sperm quality, higher maternal age, and when you are going for a pre-implantation genetic screening or a pre-implantation genetic testing. So all those indications require intracytoplasmic sperm injection. While a low number of sperm in ejaculated, that can be because of retrograde ejaculation, blockage in vas deferens. So the tube that carry the sperm to the testes is blocked. So complete absence of spermatozoa in the ejaculatory semen sample. But yes, there is a live, motile, and morphologically normal spermatozoa inside the testes. So to retrieve those spermatozoa for the ICSI, there is a specific surgical methods are used. That is called a percutaneous epididymal sperm aspiration, testicular sperm aspiration, and a microepididymal sperm aspiration. Here in this video, you can see ICSI holding needle, hold the oocyte, and with the help of injecting needle, single sperm injected inside the cytoplasm of the oocytes. So ICSI is a very precise technique, and for that you require a specific instruments, different types of media, and all those things. For ICSI, you require hyaluronidase for denudation of the oocyte, polyvinyl pyridone (PVP) for slower down the motility of the spermatozoa. ICSI holding needle, ICSI injecting needle also you require, and a different type of micromanipulator are also there. So sperm that are abnormal, usually, sorry for the disturbance, maybe there might be some technical issue. So sperm that are abnormal and cannot, madam, am I audible? Hello?
Yes, madam, you're audible.
Okay, fine. Yeah, thank you so much. So cannot swim directly. So ICSI in normal ejaculate, there are million of sperm, but they are straightforward. So ICSI facilitate us to overcome this particular problem with getting those immotile sperm injected into the eggs. But the most important factor is problem with the antibody, anti-sperm antibody. So this can be produced by in male partner in the ejaculatory fluid and in female partner in cervical secretion. For whole the IVF, you require a specific dedicated IVF OT in which only a embryo transfer, ovum pickup and embryo transfer has been carried out. ICSI workstation include a micromanipulator, citizen microscope, heating block, laminar airflow, and as madam already discussed about the specific incubator, whether it is a benchtop incubator, box incubator, single gas incubator, CO2 incubator, or a premix tri-gas incubator. in which a 6% CO2, 5% oxygen, and the proportionate nitrogen. This one is a ESCO-MIRI incubator, a multi-room incubator here. This particular incubator, there is a ultraviolet filtration system also for the gas. In this particular incubator, nitrogen and oxygen supplied by cylinder, and it automatically takes oxygen from the atmosphere. Most important thing in all IVF lab is the sterile, clean air. So for that, to overcome this, we use a LabGuard or a Coda Aero HEPA air filtration system. So in whether it is a IVF or ICSI, procedures start with the ovarian stimulation. We give the female specific gonadotropin, follicle stimulating hormone, or hormone for the follicle growth, and every alternate day we measure the follicle size, and when a maximum follicle reaches to 18 diameter, we give her a HCG injection, and exactly after 36 hours of the HCG injection, we carry out ultrasound guided oocyte retrieval. Here in this video, you can see one by one follicle aspirated with the help of the needle. And this whole procedure carried out under a general anesthesia. We use a specific ovum pickup needle, and transvaginally we aspirate a single follicle one by one. So this one is a very precise technique, and for that technique you require a specific skill, because there might be a chance you can damage artery or some damage, because it is a completely blind procedure. When follicular fluid uh, uh, inside the lab, you need to identify a specific oocyte, and there are a mature oocyte, immature oocyte, M2, M1, or the GV oocyte. For insemination, whether in case of IVF, just you need to mix egg and the sperm. And for ICSI, there is a some different procedure. You culture the embryo whether for day 2, day 3, or day 5, that is called the blastocyst. And after those culture, transfer the grade A embryo, usually one single embryo transfer or a two embryo transfer. In case of blastocyst, I usually transfer a single blastocyst. Whether in case of day 3 8 cell embryo transfer, I usually transfer a two embryo mid cavity of the uterus 1 centimeter below the fundus with the help of embryo transfer catheter. And this embryo transfer technique should be atraumatic, no any type of blood or mucus present inside the catheter, and the quantity of the fluid with the embryo is less than 30 microliter, and embryo transfer, it's a matter of only 30 to 40 seconds. So try your level best, the embryo transfer should be done within the 30 to 40 seconds only, without any trauma, without any contraction of the uterus. For the ICSI, we need to denudate the oocyte. What is the denudation of the oocyte? Denudation means the removal of the surrounding corona and accumulus cell from the oocyte. For that particular, we use a 80 IU per ml hyaluronidase solution. But 80 IU per ml, I also dilute this 80 IU hyaluronidase, because the final aim is that we denude the oocyte without any type of damage, and for that we use 140 microliter denudation pipette for the oocyte denudation. And this all oocyte denudation carried out 3 or 4 hours after the ovum pickup. That is most important thing. For ICSI, there are different type of microscopes available, like Nikon, Olympus, and the micromanipulator from there are 3 type of micromanipulator: Narashige, RI, and the Appendrock. And the specification are different from each other. I used to do ICSI in Nikon Narashige combinations, but there are a lot many advantage in ICSI and Appendorf, and that also differ from cost also. So for the ICSI, you need to fix the ICSI holding as well as the ICSI injecting people in the specific time, that no any type of air bubble present inside the needle. And the alignment of the ICSI injecting and holding needle is most important, because oocyte ICSI holding needle and ICSI injecting needle must be in a same focal plane. It is most important thing. So with the help of ICSI injecting needle, we immobilize the sperm by just crush the tail of the sperm. You must take care about that. Do not injure the midpiece or head of the spermatozoa. Just simply crush the tail, immobilize the sperm, and gently aspirate the sperm inside the injecting needle. And here the whole ICSI procedure, here you can see holding needle, hold the oocyte with a negative suction, and polar body must be either a 12 o'clock or a 6 o'clock position, and injection of the sperm at 3 o'clock position. Else you will damage the chromosomes and spindle also. So slightly pierce the oplasm of the oocyte, cytoplasm of the oocyte, and aspirate some cytoplasm along with the sperm, and slowly release the sperm inside the cytoplasm. Here in this video, you can see the whole ICSI procedure. With the help of holding needle, we hold the oocyte, and we adjust the polar body at the 6 o'clock position. Here you can see a fine zona pellucida, cytoplasm, a perivitelline space, and a polar body at 6 o'clock position. And it is most important thing that oocyte holding needle and the injecting needle must be in the same focal plane, else you lies the ended up with the lies of oocyte, or the no fertilization occur in the oocyte. So adjust all those three things. ICSI is a very precise technique. A specialization is required for that particular technique, and smoothly inject the sperm with the minimum volume of the media, with the minimum volume of the PEP. Here in the injecting there, you can see this one is the sperm. Bring the sperm near the tip of the injecting needle, pierce the oplasm like that without damaging the oocyte. Slightly negatively suction the cytoplasm, and release the sperm in a center of the oocyte. So this one is a whole ICSI procedure. Here you can see slightly negative suction of the cytoplasm along with the sperm, and then release the sperm here in the center of the oocyte. So this funnel type of picture is most important thing that indicates ICSI is performed and the sperm injected inside the oocyte. This funnel indicates proper ICSI done. And remove the needle. So this is the ICSI. So what are the benefits of ICSI? ICSI enables the fertilization to happen when there are very few sperm, oligospermia, and the severe male factor infertility. Before the development of ICSI, male infertility related issue or the abnormal sperm count, teratozospermia, globosospermia, consider it largely untreatable. So this particular assisted reproductive technique therefore enable the couple where the earlier they have no any choice, instead of using a donor sperm. Now with the help of ICSI, they have their own biological child. ICSI benefits includes helping the couple to get pregnant when the dealing with following male factor infertility like low sperm count, physiological blockage, varicocele, poor sperm motility, as well as the morphology. And previous multiple failed attempts, one or two failed attempts of the IVF, and desired pregnancy after the post vasectomy. If we consider about the results and success rate, clinical pregnancy rate, so 35% pregnancy rate in the women where the age between 30 to 35, and the 20% pregnancy rate where the age 35 to 40%, and only a 10% success rate after the 40 age. So it is better that if you know your problem, whether it is a male factor or the female factor infertility. In female factor like blocked fallopian tube, advanced maternal age, multiple fibroids inside the uterus. So you directly go for the IVF instead of wasting of years and years. Scientific data shows that blastocyst embryo transfer on day 5 or day 6 stage give a higher successful pregnancy rate compared to day 2 or day 3 stage embryo transfer. And depending upon the age of the couple, but this success rate also vary depending upon the age of the couple, their physical status, like any other disease, diabetes, hypertension, some renal or liver disease, and their lifestyle. We usually know that female with over excessive body weight or lots of stress in male, alcohol drinking, nicotine use, over stress, anxiety leads to infertility, and for that particular cases success rate is low. And IVF ICSI success rate is also depends on your lab quality, your culture media, your incubator, and individual performance, embryologist perform skill also. Uh, for it is mandatory that every week you need to check the quality, do the quality control and quality assurance of your IVF lab, and proper media gassing, eliminate the any type of contamination, proper air quality inside your IVF lab, and always follow the standard operative procedure in your IVF lab, proper stimulation protocols, ICSI, IVF, proper embryo transfer technique, and the culture of the embryo. The quality of gas, oxygen, nitrogen, and CO2 is also most important thing. And you in our IVF lab, we always use a CODA filter for gas purification, and inbuilt CODA Aero and the lab guard to purify the air quality, reduce the particle count inside the IVF lab. So there are lot many risk factor of ICSI also. ICSI has been used since last early 1990s, and preliminary studies on the children born as a result of ICSI do not suggest increase any congenital abnormality. However, children conceived through IVF as well as the ICSI uh, for the reason of severe male factor infertility may be more likely inheritance certain genetic conditions also. ICSI or IVF increase the success rate in IVF, but the procedure is always not a perfect. There are lot many manipulations is there in ICSI, denudation of the oocyte, injection of the oocytes, sperm present inside the polyvinyl pyrrolidone. All those factors affect the final fertilization rate and final embryo culture, embryo gradation, implantation, and pregnancy rate. It is not like that if you do the ICSI, your all embryo will get fertilized, damage to the embryo. Fertilization rate with ICSI is range from 50 to 80% only. That depends on quality of the sperm and the oocytes and other chromosomal abnormality of the sperm and oocyte, DNA damage of the sperm also, ROS factor also. So not all fertilized eggs develop into the healthy grade A embryos. Unfortunately, some eggs are lysed also. Becoming a pregnant with ICSI or IVF give a multiple pregnancy. Since ICSI take place, there is a chance of multiple pregnancy. There is a chance of ectopic pregnancy also. The scientific data shows that couples that use ICSI with IVF have about 30 to 35% chance of the twins pregnancy, 5 to 10% chance of having a triplet pregnancy. So carrying a multiple pregnancy also increase the chance of your antenatal workup like gestational diabetes, high blood pressure, low amniotic fluid. Uh, it require bed rest. It also, if it is a multiple pregnancy, that patient requires cesarean section, preterm labor, and you need to do a cervical circulars to prevent the preterm labor also. Here, this is a video of embryo from embryoscope. You can see here in the video blastocyst formation, a fine zona pellucida, and grade A blastocyst. It again increase the number of cell in, and finally it hatch and all the trophectoderm cells come out from the here. So this one is the fine video from the embryo scope. Some specific risk factor related to the birth defects, child born from the ICSI. So the sex hormone abnormalities, hypospadias, a birth defect in specific into the boy, and Angelman syndrome, Beckwith-Wiedemann syndrome also scientifically reported while the child birth with the ICSI process. So, my take-home message, and according to the different concurrent scientific study, there are no studies showing that the ICSI is beneficial where poor quality of the oocyte, low oocyte lead, and the advanced maternal age. ICSI cannot increase the improve the live birth rate in all of those cases. Of course, ICSI appear to improve the fertilization of the oocyte, while oocyte undergone through in vitro maturation. ICSI, also, ICSI is beneficial on a cryopreserved oocyte. Because of cryopreservation, vitrification, or the slow freezing, zona pellucida of the oocyte become more harder. So ICSI is beneficial compared to the normal IVF. ICSI can increase the fertilization rate when lower fertilization or a no fertilization in previous IVF cycle. In cases where without male factor infertility or a history of no fertilization, routine, I do not recommend ICSI for the all the oocyte, whole ICSI cycle. ICSI is usually done if it is indicated, not ICSI for all. ICSI is also useful for pre-implantation genetic testing, pre-implantation genetic screening in the absence of male factor infertility. Thank you so much for patience hearing, and the session is open for a question and answer.
I request you to pass on the questions to Madam.
Yes, ma'am. Good afternoon, everyone. I'm Shweta. Ma'am, there are a few questions from the audience. So may I
Yes.
proceed?
Yeah, sure.
So the first one is, in the view of cost effectiveness and success rate in art, which one is preferable, ICSI or IVF?
Specifically, if it is not indicated, I prefer a IVF because oocyte naturally select the normal spermatozoa, right? So I basically prefer a IVF. But if it is indicated, you need to go for the ICSI if it is male factor infertility or previous failed attempts of the IVF cycles. Or if previous cases, you already know that there is a very hard and darker zona pellucida.
next one is, does ICSI increases the risk of autism spectrum disorders?
Not yet. It is a questionable. It is a questionable because there is no much study going on in this type of particular. Only a chromosomal some abnormality and some chromosomal disorders have been studied in some extent.
Ma'am, and the next one is, during this procedure, is there any chance of damaging the nucleus of ovum and how to avoid it?
Yes, there is a chances of nuclear damage the nucleus of the ovum. Nowadays, a spindle imaging system, spindle view is also available. Laser assisted ICSI is also available. So, if you are have enough skill, so always pierce the oocyte at between the 3 o'clock position, and your polar body must be at 12 or 6 o'clock position. If you have a higher end technology like spindle imaging system, so you can do your ICSI along with the spindle, so you exactly know where the spindle is located. So no need to damage the spindle. If you damage the spindle, your oocyte will be lyse or no fertilization take place. You directly damage the nucleus.
Okay, ma'am. And ma'am, why some oocytes degenerate after ICSI?
Oocytes somehow a higher concentration of the hyaluronidase also. Second thing, your vibration of ICSI table, because for ICSI you need to have a anti-vibration table. So your concentration of the PVP, there are multiple concentration available, 7%, 10%. Hyaluronidase also a different concentration, and your scale of the ICSI. If your oocyte and your injecting and holding needle are in not same focal plane, you are going to damage the oocyte or degenerate the oocyte.
Okay. And ma'am, what about the chromosomal aberration?
Chromosomal aberration, if you use the spindle imaging system and if you know the particular site of injection, so there is not for that you also do a PGD and PGS also, taking a single blastomer after the ICSI.
Ma'am, and can hydrocele affect male infertility?
Not yet.
Okay. That's it, ma'am. Thank you.
Yeah, thank you so much.
Over to you, Mini ma'am.
Thank you, Shweta. Thank you, madam, for such an informative talk on ICSI. Thank you, madam.
Thank you so much, ma'am.
Now I welcome the most awaited speaker of the day, Dr. Kersi Avari, founder and director Embryology Academy of Research and Training at Mumbai. Sir has been practicing embryology since 1991 and is a program director for one of the most reputed embryology training institutes. Sir is PhD in reproductive endocrinology and a postdoc in andrology. He is a recipient of 2 fellowships, the Lady Tata Memorial Trust Junior Research Fellowship and the Golden Jubilee Scholarship by Institute of Science, Mumbai. Sir has done extensive work with various steroidal contraceptives on various animal models. He is scientific director at ART, which has been endorsed by 2 international brands, the Center for Reproductive Medicine, Cleveland and Clinic, USA, and Embryology and PGD Academy, London. Sir has been offering embryology and allied application support services to various IVF centers in India and abroad. More than 20 publications to his credit in national and international journals. The list of contributions in the field is endless. Hence, over to you, sir, to enlighten us on the topic of andrology, the first stepping stone towards actual understanding and implementation of techniques.
So, good evening to all of you. And of course, I thank my 2 prior speakers also, Dr. Pratiksha and Dr. Doel. And I appreciate their cooperation into this project at a very short notice. But before I start, let me thank the MGM Institute of Health Sciences, especially the Honorable Vice Chancellor, Dr. Shashank Dalvi, Honorable Pro Vice Chancellor, Dr. Nitin Kadam, Dr. Mansi Thakur, the Director, organizing secretary, Dr. Minnie Mole, and all my student friends and colleagues who have tried their level best to make this presentation and make this program a grand success, and it rolls smoothly as it looks like. Well, the two prior topics made us know about the intricate procedure, that is the intracytoplasmic cell injection, that's the ICSI. And the second one is the actual role of media and the culture techniques, how efficient and how important they are, and how much do they prove as a backbone of embryology. But now this topic which I'm trying to take is andrology. But why andrology? Why is it? It's the first stepping stone. If anyone who wants to introduce himself or herself into the field of embryology, this is the first thing that they have to learn. The reason is they get the chance to work and to handle the actual gametes, which are comparatively much easier to be available and much easier to manipulate in the laboratory. Because every infertility specialist also would dream of starting his or her own laboratory setup. Every budding embryologist will also dream to progress further and further. And for all, the first stepping stone towards a successful endeavor in embryology is via andrology. So andrology was a medical specialty which deals with medical health, relates to the problems of the male reproductive system, urology. Initially, it was just urology. Subsequently, when the interest in male infertility blossomed over the years, it started getting diversified or a bit channelized. And there were certain urologists who started focusing their attention towards the male reproductive system, the problems, the anomalies, the difficulties, how they could be rectified, how they could be set through. So now we have got even dedicated journals totally devoted to andrology. Now we know that as far as ART is concerned, the two pioneers in their tiring efforts, Dr. Edwards and Dr. Steptoe, we all know the first IVF baby, Louis Brown. We also know the first baby from the cold, or what we call it as cryo-preserved embryo, subsequently thawing. And then the first ice baby was in 28th March 1984. Now, since then, when small labs, a basic IUI lab, or a basic infertility specialist who wants to hone her skills, and if she doesn't want to go into an intricate or the second or the third step, first what was followed was a very basic holistic approach, what we call as timed intercourse. Now over them also, the gametes of the males could be seen easily as they are collected, as they are assessed easily, what we term as semen analysis. But in the lady or the female, the only modus operandi by which we can monitor them is by hormone estimation or by ultrasonography. We see the follicle growth, we see the follicle pattern, we see the ovarian content, etc., etc., etc. Now, this timed intercourse also had a basic parameter, or the most important yardstick of a success of a timed intercourse is also depending upon the number of sperm. The quality, quantity, and the other various diagnostic approaches I will take in this lecture. Now, you know there are three basic procedures, IUI, IVF, and ICSI. In all, sperm are essential. Sperm are assessed, sperm are processed, sperm are cultured in the ways verbatim same in all these procedures. That's why whenever anybody says it's a sperm processing lab for IVF, I say no, it is a general sperm processing laboratory, whether you do an IUI, whether you do an IVF, or whether you do an ICSI. The yardstick of quality control remains the same in whatever it is. Now, well, first discovered in 1677 by Antony von Leeuwenhoek, and it was supposed that it's a small animalcule with the whole human content which was nestled beautifully in the head, what we subsequently today call as the acrosome. Now, the same gentleman was also responsible for improvisation of microscopy. Now, this is a detailed version of the human sperm with all the proteinaceous parts, the proteins, the acrosomal proteins, the midpiece, etc. This is more into a mole biological approach. But this presentation is made in a little simplified version, especially for the budding embryologists who would like to venture further into the field and who would like to have a gradual step-by-step introduction as to how to assess the sperm and as to how quantify the male as far as his androgenicity is concerned. Now, for a layman, the sperm is divided into three parts: the head, midpiece, and the tail. The head consisting the genetic package, midpiece the mitochondrial sheath, the energy source, and tail locomotion. The three parts, however different in function, but for the total integrity, or what I would say as a functional integrity of the sperm per se in general, depends upon all these three parts working in unison together for maximum fruition. That's the reason one parameter called morphology is the backbone of semenology. Now, a little bit of morphometry and its kinetomatics. Now, I would say sperm are present. Well, they are present. They are perfect in their dimensions. They are perfect in number. But what if they are not motile? What if the kinetomatics is at a very low grade level? Now, in such cases, you have to give a proper diagnosis, a proper future way of recommendation as to which method subsequently would be effective for the poor childless couple. Because remember, an infertile couple is mentally, physically, and financially stressed out. They would just say at the end of the day, the doc, please do anything, but I want a pregnancy. Now it is for us how to quantify, how to qualitate, how to report, and subsequently how to diagnose the problem and then select a technique which would be quite appropriate for us to get a result. Now, embryology is a dynamics between two gametes, excuse me, the oocyte, which is the largest cell of the body and the most sensitive cell, versus the human sperm, which is the tiniest and the most motile cell. And it doesn't mean that the fusion of these two can effectively yield an excellent embryo. No, there are different time-related, stage-oriented procedures or steps which it has to go till you get an excellent 3-day morula or a 5-day blastocyst worthy of implantation into a well-receptive and a well-primed endometrium or the uterine environment of the lady. So this, I hope you have seen the details on the screen, because when a sperm moves, the tail, whiplashes, effective stroke, recovery stroke, where does it get the energy? Well, there is a fantastic mitochondrial package, mitochondria, the powerhouse of the cell, ATP, propulsive moment, active, moderate, sluggish, the yawing, the thrust, the torque, etc., etc. It is dynamics. Now, sperm dynamics can take up a time more than hours as far as deliberation and discussion is concerned. But what we have to see, or what a skilled andrologist or an embryologist or a seminologist would see, is within one glance, on a good bench microscope under 10x and 40x, he has to check out the sperm quality, their locomotion, their yawing, their propulsion. Of course, you can't make out the morphology in a wet specimen, but still some large morphological disorders like globoids, bent necks can be detected. Now, this is the basic, the raw material or the source, the seminiferous epithelium in the testes. This is a tangentially cut section with a hematoxylin eosin, and it shows you a number of seminiferous tubules. Now, this is the production site. The testes situated in the scrotal sac outside of the body temperature. The seminiferous tubules, if drawn out, can extend into meters of length, but they are so nicely and compactly packed together that they keep on proliferating, because this tissue is a germinal epithelium. The male and the female both have a germinal epithelia as far as production of the gonads are, the cells are concerned, because they are remaining in a dormant condition from the birth till the puberty comes in, and there is an activation of hypothalamo-pituitary gonadal axis, which literally gives them a kickstart. And they as if wake up from the slumber, and then starts the changes, and then starts the induction or the introduction of the secondary sexual characteristics in an individual, a male boy or a female girl. And then we can say, oh, they are sexually mature or sexually active. Now, before the hormones started, or before the radioimmunoassays started, what we used to do were just testicular biopsies. We used to cut open the scrotum, pinch out a tubule or a chunk of a testis material, and we used to fix it in a special fixative, which is Bowen's fixative. Because in fixing of testicular tissues, epididymal or reproductive tissues, which are very soft and sensitive, we prefer Bowen's fixative, which is a mixture of picric acid, glacial acetic acid, and a little bit of formalin. Because unlike absolute formalin and absolute alcohol, picric acid doesn't shrink the tissue, because if the tissue gets shrunk, then the histopathological inference would be improper. So we used to stain them, we used to cut them, and then there was what we call as testicular score. It was like Sertoli cell syndrome only, or normal spermatogenesis. Now we used to count 100 tubules, and out of which 60 tubules would have been showing, 60 tubules usually show a very good amount of spermatogenesis. See, these are the spermatogenesis, this is the lumen in which the spermiated sperm collect. So when the 40 tubules which were in a resting phase, they subsequently in the next cycle of 70 days, which is one spermatogenic cycle, they used to be active. So then there is a rotation. Now the question is that these testes are in the scrotum outside of the body cavity at a less temperature, 1 to 1.5 degrees less. But once they are ejaculated, they have to be collected and stored exactly at 37 degrees, which is the activation temperature, optimal activation temperature for the sperm from vivo to vitro. Remember, hypo and hyperthermia both are bad for the sperm. There is just one terrestrial mammal who has the testes in the body cavity. That is the elephant. The elephant has the testes in the body coelom. It is insulated by layers of fat. So this is basically, now anatomically also there are lots of investigations to be done. Undescended testes, improper form testes, spermatogenic failure, testicular cancer, subsequent hydrocele, varicocele, all can affect the testis sperm production. Fever or hypothermia. Even a person having high fever, it is always suggested, he is never recommended a semen analysis for another 2 weeks. Now, this is exactly the scenario what goes on inside. You see the lumen, you see the battery of the germinal epithelia, spermatogonia A, spermatogonia B, primary spermatocytes, secondary spermatocytes, 3 types of spermatids, early phase, cap phase, maturing phase, and the lumen into which the small freshly spermiated sperm collect. And during the excitation phase or the orgasmic phase, when they collect, they go towards the rete testes, collect in the caput, subsequently transferred to the chorda, and by the muscular contraction during orgasm, they are ejaculated all the way from the vas deferens through the accessory gland complex, and they come out through the penile opening, which is the same for urine and semen, which we collect as a sperm ejaculate. Now, for developmental biology, this is just an idea. Which derm is responsible for formation of the entire gamete and the accessories? We know we have got ecto, meso, endo, ecto and meso from somatoplore, meso and endo from splachnoplore. There are division of cells, there is cell migration, there is cell accumulation, then there is organogenesis. The list is endless. Now, how we quantify male infertility? Well, there could be spermatogenic problem and there can be erectile dysfunction. In a spermatogenic problem, the person is active, he has a fantastic erection which stays long. He can have a satisfied sexual act, but the content of the ejaculate, which is the sperm, is too abysmal for his partner to get a pregnancy. Whereas the vice versa in case of erectile, it's a psychosomatic stress disorder, a vasomotor symptom or nervous disorders, problem, lumbar area. So an erectile person can be producing sperm, but unfortunately cannot consummate the marriage, whereas a spermatogenic disorder has the battalion of sperm ready, but unfortunately can't justify because he is either oligo, severe oligo, necro, astheno, or OAT. Now, in a nutshell or in one line, I have just written what an ideal sperm is and what an ideal sperm is capable of doing. Now, a sperm acrosome is like a glorified lysosome. Once it breaks the oolemma, does the membrane reaction, enters the cytoplasm of the oocyte, then there is decondensation. Its nucleus is liberated, which again divides. Then one part of it forms a polar body. One polar body is already present of the egg. So 2 polar bodies and 2 polar nuclei tend to form the zygote, which is nothing else but the first step towards an uncleaved embryo. Now here I have written angle of yawing. Now, you know, a sperm moves like this. It doesn't move just like straight. So yawing is a phenomena in which a body tends to travel in a straight line, but one part, that's the head, always keeps on changing the angle. There can be negative yawing, which is useless. Yawing is supposed to play an important part when the sperm approach a mature oocyte that is metaphase 2, having a lovely composition of its granulosa cells or the cumulus cells. There they form the reaction hyaluronidase. They eat up the granulosa cells, clear the way for one sperm to go and do the membrane reaction, which ultimately results in a pregnancy. So this is exactly a pooled effect of something like 30 to 35,000 actively capacitated sperm with fantastic progressive motility and linearity to form a healthy zygote, which subsequently down the tract of the system in the tube in different areas, whether it's an isthmus, whether it's the mid part, step-by-step developmental progression takes place in the tube, which is nothing else but a glorified incubator. Now, this will tell you what happens in a day-to-day basis, hour-to-hour changes. And what we are doing in the IVF lab, we are basically trying to mimic the same scenario. Now, over here, people say, what is the role of the sperm? Well, a well-capacitated, well-processed sperm can definitely increase your chances of success in any ART procedure by as much as 15%. Remember, we can never exactly mimic the vivo to vitro conditions because for a successful pregnancy, there are listed like 154 conditions, out of which we can have a control only over a minimum of 30 to 35. So when we are getting even say 30% or 25% pregnancies, let us not get discouraged. We are trying our level best because getting a beta HCG positive at the end of the day after doing a procedure, that is not the thing. The beta HCG has to be repeated after 2 days. There has to be a quantum increase in its level, or I could say a geometric increase. Then what's the next step? Cardiac activity. The cardiac activity will tell us, well, definitely there is a fetus in the live fetus in your uterine cavity. And then the ultimate test, the take-home baby rate. So there is quite a distance between the cup and the lip. So the initial beta HCG results have definitely to be taken with a smile, but they are not the end of the story. The race towards a successful pregnancy has still begun. Now over here, when we are doing ICSI, as Dr. Pratiksha suggested, when we are doing ICSI in a poor sperm sample, we have to find the best amongst the worst, which is quite difficult. Egg quality, because everything in embryology which is being done is done by morphometry. There is no biochemical method, there is no mole biological method like our routine blood assays. We can judge what the condition is as of now. Now this makes the sperm part very challenging and interesting. Now these are the three things. In ART, what you have to be careful is recovery, survival, and viability. You recover the gametes, you make them survive, you make them viable, so at the end of the day, they are there to give you a successful result. Now, WHO Semen 2010 criteria, it's easily available on the net. You can just download it. Now, these are the reference values down the years. Now, majority of the parameters same are well quite within the range. But if you see the normal forms, in 87, it was 50. And in 2009-10, that percentage has dropped to a mere 4. Now, this is a monumental drop. You know, it's a monumental drop. Now, this is a challenge. That remember, always quantity and quality don't go hand in hand. This has given rise to the embryology scenario, venturing into different modus operandi techniques as to what shall I do to select the right sperm. But believe me, till today, we cannot say with an authenticity that we have selected the right sperm. No, you can't say that. There are many techniques available. There is IMSI, when you do an ICSI. There is intracytoplasmic magnified sperm injection, which can magnify the sperm to as much as 7,000 times. There are DNA fragmentation tests, which say, oh, the DNA was fragmented, so this sample has a high rate of sperm showing this, so no, it cannot work. But still, sometimes luck favors not only the brave, even the stupids. So what I'm trying to say is we are trying our level best to not guarantee results. We are trying our level best, excuse me, to optimize the results. Now this you are all aware of, the frontal view and the side view. We are sure that once produced in the testes, the sperm move towards the epididymis, 3 parts individually called as epididymids, caput epididymids, corpus and cauda. In caput, there is maturation, effects of enzymes, glycerophosphorylcholine, sorbitol dehydrogenase, lactate dehydrogenase, alkaline phosphatase. So the sperm get a little stronger. Then they are traveled all the way to the cauda. Cauda is a storage. So in cases where there is a vasa block or a blockage in the vasa deferens, what will the andrologist do? He will just take a needle and just pierce the epididymis, what we call it as PISA, percutaneous epididymal sperm injection. You get small little frogling-like structures wiggling along because the sperm from cauda will be 15 days younger than the ejaculated. The sperm from the testes, the testicular aspirated sperm, will be 21 to 23 days younger. So there is a route: produce, mature, store, transfer, ejaculate. It's a procedure which depends upon various stimuli: orgasm, tactile, sighting. Now the problem is one semen collection ideal for us to quantify a person? No. There are lots of factors, psychological factors, traumatic stress factors, which a gentleman will be induced in when he's undergoing the procedure. You can't say, it's not like blood, you take a hypodermic, push it in, pull out the blood and see whatever it is. The gentleman has to be in a mental state of mind. Otherwise else, a perfectly normal person under stress, when he is told to ejaculate in a collection room which is absolutely new to him, he will be suddenly undergoing into a tension or a stress. So these are called inhibitions. We have to break the inhibitions. And one semen sample doesn't a summer make. Collect 2-3 samples with a gap of 15 days and an abstinence period of 3 days prior to each collection, because an abstinence period will ensure that the sperm reservoir is filled back to its capacity once again. So depending upon the volume, depending upon the appearance, depending upon the pH, a semenologist can judge what is wrong, or an andrologist can judge what is wrong. Now these will be the secretions, because when there are different phases, excitatory phase, ejaculatory phase, and a flaccid phase. When an excitatory phase is there, the bulbourethral glands will give out the small secretion in very less amount. They are always acidic. And what do they do is, they clean the pathway. I'll just go back and show you, sorry. Yeah, they clean the pathway as through the penile area and remove it of old urine residue. So it's like a it's like a pilot car which goes ahead. So if a collection is there and this is just 0.3 ml, which is glassy in color, which is acidic, the patient will say, sir, collection ho gaya. No, it's not like that. So what we have got to do is we have to first counsel the patient properly. Then the prostate will suddenly spring into action, pump itself, and the prostatic secretion will come out with a little bit of sperm. And then the first squirt which will come, which will contain at least 40 to 45% of the chunk of the spermatozoa. So when the patient collects, we have to counsel him that the first squirt of the ejaculate contains the chunk of the sperm. So please see that the first squirt doesn't go waste. Unknowingly, what did the poor patient say? Sir, first part I spilled, but the last part I collected. They have a feeling that the last part consists the chunk, but no. The last part of the ejaculate consists of the seminal vesicle secretion, which is the maximum. And it is alkaline. So ideally, the pH of the semen is alkaline between 6.5 to 7.5 or a little more, because 80% or 85% of the semen consists of the seminal vesicle secretion. Epididymal and testicular secretion is quite less. There will be number of cellular inclusions. Dead sperm, alive sperm, abnormal sperm, premature cell, pus cell, epithelial cell, bacteria, debris, granular, cellular, phagocytosis. So semen is a plethora of flora and fauna. Infections, yes. There is a margin or a barrier beyond which it is toxic, beyond which it is cumbersome, because once the infection comes into the washed semen sample, it can cause multiple problems. It can cause multiple problems in the culture dishes in which we inseminate. Though in the ICSI we just take one sample, but the peripheral contamination of the microbial content adheres to the sperm membrane, because sperm membrane has a fantastic power of adhesion. When a person suffers from HIV, the sperm are not to be blamed. Please, it's the contaminated secretion of the prostate and the seminal vesicles which coat the sperm, and the HIV virus get adhered to the membrane. Well, three another things we have to recognize. Many a times gentlemen complain burning sensation whilst collecting, frequently voiding of urine, feels like going off and on, content is too less. He may have prostatitis, inflation of the prostate, or the symptoms are mimicked as UTI also, urinary tract infection, lower. Simple. What to do? Get a urine examination, get a semen examination. If pus cells are there in the urine, chances of a UTI is high. Pus cells are there in the semen, prostatitis. Second thing, we get sometimes large ejaculates, huge, 7.5, 9 ml. Well, nothing to be proud about it. These secretions will be slightly tinged with yellow. And the sperm content in there will be there, but the sperm will be devoid of active motility. Now remember, seminal vesicle and prostate are both secretory glands. There are tubules. They are lined by tall columnar secretory epithelial cells. So whenever there is an infection, they will secrete copious amount of fluid to wash off the infection, as a result of which the volume will be large. But the semen containing seminal vesiculitis secretions will be containing a higher amount of bacterial load. So if you do a 24-hour sperm survival test, the next day will be a plethora of beautiful bacteria wobbling inside your culture. The third option is hemospermia. When there is a reddish color in the semen, it's a sign of danger. Prostatic carcinoma, bladder carcinoma, or a severe bladder infection. That patient has to be immediately sent to an andrologist for further consultation. So these are the ample things. A good collection room. Please, I have seen so many labs where the collection room is just a small room, and inside it is utilized as a storage room, as a changing room. And many places, I would say, sorry to say, with regards and respect to all of the doctors who might be on this webinar, the collection rooms are simple toilets, which are the biggest turn-off for the patient. The patient is under stress. He doesn't know what to do. He is confused. So therefore, the latest ICMR guidelines have also mentioned that whether be it a primary, secondary, or a tertiary center, please, I would want the lab to have a good collection room, a homely ambience, a cot, a couch, a TV, some erotic literature, wash basin, no soap because gentlemen use for lubrication and soap is toxic because it's alkaline. A small washroom, sterile tissues, and tell them that for half an hour, there will be a no knock sign on the door. But if your collection time increases by 30 minutes, I will be the one who will be knocking, because if a gentleman is not able to do the collection in the first 30 minutes, he will never be able to produce a sample. We will force him, try again, he'll get exhausted. And then what you tell him? Oh, now we'll do a tisa pisa. Now you can't do that because the epididymal musculature has gone flaccid. You can't get the source to put your needle in. So in spite of being perfectly normal embryologically, psychologically we have made him abnormal. Okay, so this tells you, some people have a trauma. No, I will never be able to collect here. I will be always ready to collect at home. Give him the container, but please give him your lab container, which is a sterile container. Individually wrapped sterile mouthwash container. Nowadays, we are getting those bio-friendly plastic containers. Initially, we were getting glass containers, but they were the best because glass is a super cool liquid. And no cell secretion can interfere with glass. So I would just request you to go through this. As I told you, spillage. First cohort contains a large percentage of election, motile sperm. Now, lastly, some have collection issues. So for them, we can give a small vibrator or an electroejaculation. Now, once you collect, you do an initial assessment of the sperm. So we see the volume. It comes to know how are the glands secreting. We see the nature. If it's a viscous one, well, there is a gland infection. pH, bulbourethral gland acidic, prostate is acidic, seminal vesicle is alkaline. Color, hemospermia, internal organ damage. Count and motility, well, you know, motility to move, kinetomatics, and other inclusions. Normo, oligo, astheno, terato, necro, azu, oligo, astheno. These are the categories. There is one more, aspermia, in which there is no semen collection. A gentleman just can't collect. Either he is having a problem or he is psychologically in an absolute depressed state of mind. So this can also work out. You have a procedure. The lady is stimulated. She is given her injection. She is about to rupture within 2 hours. You are going to do an IUI, but my God, you don't have sperm. What to do? There are stressful conditions. 5 to 10% of males experience difficulty in masturbation. Some just can't masturbate. Some can masturbate and collect and ejaculate, but some can't ejaculate during intercourse. There are lots of confusing problems. The thing is, we can request them to do coitus interruptus, but in coitus interruptus, majority of the time, the first quart of the ejaculate is lost. Counsel the person and remove the apprehension. Some of them are taught, no, never to masturbate. Oh my gosh, it's against. It will ruin your health. So all these things play a havoc on the mind of the person, because that person all his life poor, has never masturbated, and when he's going for a treatment for infertility, we are telling him, okay, masturbate and get a sample. So there are unrealistic expectations from us. There is stress. Proper collection room is not there. Time factor is there. Our demand is there. Look, tomorrow your wife is having a procedure. Please, I want a good sample tomorrow. Please, please, please. There is a battlefield of thoughts in that person's mind. So what to tell him? During the peri-ovulatory period, tell him to try masturbation. Whenever he collects a sample, freeze the sample. Give him a Viagra support for erectile reassurance, or you can give him a vibrator to gain confidence. See, these are the reasons. No privacy. Sitting in a room, the sister shouts, Mr. So-and-so, collect the sample. Oh, that's rubbish. You can't do that. Collection room is not proper. It's a changing room, come storage room, just a bench or a couch. No, no, that's not proper. Our approach is over demanding. Please collect. So what you should do is, a person needs assurance. A person needs counseling. Or last, when everything fails, tell him, boss, go home and collect and get it. But remember, we take a consent, because many a times what had happened, there was switchover of samples. So Mr. and Mrs. blah, blah, blah have done the procedure of an IUI with the sample collected from home, delivered to the lab on such and such a date, signed by both the wife and the husband in presence of each other. Now this is a vibrator. It's a simple gadget. It's like a body massager. With persons having initial erectile problem or a little difficulty in collection, what is done is a person is made to read some erotic literature till he gets a semi-erection, and with a semi-erected penile, this part, the base is kept over here, and then the machine is switched on and the machine vibrates. So the lower part or the shaft, not the head, is mostly richly innervated with nerves. So that vibrates and that gives him an orgasmic effect. And at the same time, he's fantasizing about sex. Believe me, this has given very good 70% of success, but make the patient practice. On the final day, just don't thrust the equipment in his hands. Come on, now you practice. Some of them are so comfortable that they literally are saying, sir, yeah, this machine is there, no issues. So even in the lab, because I always prefer a sample collected in the lab rather than collecting from home, because the time factor comes into play, the way they bring it, the heat factor, multiple factors are there. Remember, sperm are also living cells. They are suspended in a media. What is media for the sperm? Here the media for the sperm are the seminal and the prostatic secretions. Now this is an electroejaculation. This is a shaft or an anal probe inserted. I'll show you in the next figure. Slowly the voltage is turned up, but it's a difficult thing under GA and under observation. It goes in through the anal opening and it reaches the area where it just vibrates or gives a vibration effect towards the prostate and the seminal vesicle. And what it comes out is an ejaculate. This ejaculate has to be taken and cryopreserved. Mostly ICSI is done in this because you can't afford to repeat this off and on. And somehow, for no rhyme or reason, it has been documented that the sperm collected post this method of electroejaculation are always a little poor as far as kinetomatics are concerned. Organic problems. Fibrosis of prostatic fossa, lumbar sympathectomy, autonomic neuropathy, urinary tract tuberculosis. So first is let him get examined by an andrologist, if possible do a surgery and then counsel him. Most important, retrograde. Just like a sphincter in epiglottis connecting the esophagus, excuse me, and the trachea, there is a valve which connects or which prevents the mixing of the urine and the semen. So when the urine comes, this is shut, bingo, the urine comes out. When the ejaculate comes, this is shut, the ejaculate comes out. But when the malfunctioning of the sphincter is there, the sperm rotates back and goes into the bladder. So a person feels that he has ejaculated. the person feels that he has ejaculated, but unfortunately he can't get anything out as an ejaculate. So there is a remedy. Now these are the reasons. Okay. Now I can tell you, yeah, how to recollect the sperm is, you tell a person to fantasize about sex. He will masturbate. He will say, sir, there is no collection. Let him wait for 10 minutes, collect all the urine, centrifuge the urine into all multiple tubes. You will see the pellets, pull all the pellets, discard the top centrifuge part, the top supernatant, immediately add media, check under the microscope. You will get a number of sperm which were present in the retrograde. Only ICSI can be done, because unfortunately, even here, the sperm have lost their kinetomatics. Now, cryptozoospermia. Crypto means hidden sperm. Now, those who are having oligoasthenoteratozoospermia or the OAT, one semen sample, one report will say no sperm. Second will say no sperm. The third will say occasional motile sperm. Well, they are all right, because in such cases, the frequency of the sperms coming off and on is not that high. It will skip because the seminiferous tubules is long. One part suddenly shows a spot, and at that time, on that lucky day, that ejaculate will show presence of sperm. So that doesn't mean that the others were wrong. So what's to be done is, we have to centrifuge the sperm, the semen, check the pellet. Amongst the pellet, you will find 1 or 2 hidden sperm. And one sperm presence means you can never tag a patient as azoospermic. Never, ever. You have to write a report very skillfully that no presence of sperm in the fresh ejaculate, nor the centrifuge residue. Kindly repeat the procedure to confirm the findings. Now, this is how the sperm would move, the amplitudes. Howsoever a sperm appears moving straight, it always goes up and down. And this is known as the lateral amplitude. The more it deviates from the main line, the poorer is the motility. Now, these are the certain velocities. The upper part shows detailed velocities, which is done by CASA, computer-assisted semen analysis. And the lower ones are the normal method under a bench microscope. Now, the latest technique tells us two things: progressive and non-progressive. Initially, there were 3 paths: active, moderate, and sluggish. But for a fruition, as far as IUI is concerned or an IVF is concerned, as long as the sperm shows A and B grade of motility, you are able to go through with those 2 methods without any hesitancy. Yawing, as I described, one part moves. Well, here a little bit of chemical properties I have stated in detail, because a sperm respires anaerobically. So glucose, fructose, and citrates are the chief secretions of the seminal vesicle and the prostate, apart from zinc. Zinc is a basic barometer for the prostate. Correct? Now, liquefaction. That's the nature. Semen comes as a coagulum from the prostate. Once collected in a lab, it should liquefy within 15 to 25 minutes. It's like inverse of spin bucket seen in a female cervical mucus. In a female, the longer the threading, the fantastic is the spin bucket. Whereas in a semen, the longer the threading, the poorer the semen. It is viscous. Because the prostate specific antigen is not strong enough to liquefy the coagulum secreted by the seminal vesicles. It is slimy, oilish. So it is going to be very difficult when you are processing the sample because you have to mix the media. It forms a sort of an emulsion, an interface, which doesn't allow the sperm to swim up. Now, basically, when we say seminal plasma in its vaginal environment, in a normal condition, it is a blessing. But once you collect the semen and keep in the lab, in the incubator or on the hot block, the same seminal plasma is toxic. So remember one golden tip. Post collection, never allow the semen sample to stand for more than 30 minutes. The damage done at a mole biological level will reduce the fertilization potential of that sperm to as much as 95%, though the motility will be there. So for a procedure, once the sperm are collected, within 15 minutes post liquefaction, process the sperm and utilize them. Whereas in natural condition, because of the hostile acidic nature of the vaginal secretions or the vaginal mucus, the amines which are present in the semen or the seminal plasma, they shield or they protect the DNA from the acidic denaturation. Now look, what an irony. One is excellent on this side, one is a little hostile on the other side. So as I told you, please get a delivery to the lab immediately if it's a home collection, collect the time in the lab receipt and subsequently judge the parameters. Now we come across to cervical mucus, carbohydrate rich glycoprotein. So we were doing the Ferning test. It was an old test. I don't know whether how many gynacs do it still today because the chief constituent is NaCl with a 99% conglomerate of water. Now this cervical mucus is substituted in our embryology labs by what we call it as sperm wash media. The sperm washing media is nothing but a saturated solution of salts, no carbohydrates or proteins. That's why it is substantiated with Heepi's buffer in order to maintain its pH integrity. And since it is Heepi's, it does not need an advanced high-tech incubator like a carbon dioxide incubator. It needs a plain simple incubator. But if an embryology procedure is there, when you have IVF media, when you have fertilization media, when you have growth media, advanced growth media, which contains carbohydrates and proteins, well, there is no other option but to have fantastic regulated CO2 incubators. But here it is maintenance of one gamete only, the sperm. So what we need is a lovely shield of EDTA plus Heepi's surrounding it to buffer it from the osmotic stresses. Now we do a washing of the sperm. In a normal condition or in natural intercourse, who does this washing? Cervical mucus. Mucus is the best filter. From the time the sperm are deposited on the external os, they get in contact with the mucus. So that's the lubrication phase for the females. So the better the lubrication, the good quality mucus, the estrogenic mucus. Estrogenic mucus means good ovulation. Good ovulation means good oocyte. So see, this is how the connectivity links between each other. Now we remove seminal bacteria. That's why we call it as wash, capacitate, select the best quality of the sperm. So that's the reason we call it as a sperm harvest. So what does a sperm wash do? It harvests the best quality of the sperm. It may be a fraction, but the ones which are best in kinetomatics, ones which are best in linearity, comparatively good in morphology, they are the ones who are recruited. So this can be also termed as a recruitment of the best quality of the sperm, whether you do an IUI, IVF or ICSI. So what happens during that time? It capacitates. Now what is capacitation? Functional maturation of the spermatozoa. Now a sperm acrosome is also like an onion, number of layers. As one gets ripped off, acrosin reaction, this reaction, Hayes reaction. Now glycoprotein layer is removed, the sperm gets activated, concentration of ions. So the activation sperm moves like this, slight improvement of the shape. The sperm is a pyriform shape like a duckbill. So all these things are facilitated so that we expect that the best quality reaches all the way to the tubes. And why are we doing an IUI? Simple. Harvest the best sperm, transfer the sperm all the way into the uterine cavity, bypass the cervix and the vaginal wastage, and inseminate them at a time when the rupture takes place, so that the battalion of the sperm from the neck of the tube suddenly gets activated, captures them, denudes them, fertilizes them, and bingo, the procedure starts. Now, this is a simple swim-up method in which we eliminate the sperm. Now, this swim-up method and the density can itself take an hour of a talk. But anyway, I'll be just cutting short. We assess the sample, we check it, we dilute with the media, centrifuge it first, remove the supernatant, add more media, wash the pellet again spin it, discard it, layer it, and see the best or a very good quality of the sperm swim up, and we take the top aspirate. So this is the normal routine gold standard of sperm separation. Okay, dilute the seminal plasma, assess the sperm late first. The first wash will remove the seminal plasma, and the second wash will do is clean the pellet of saturated chemical impurities. Now the next one will be the density gradient. It's like two layers, two gradients. I put the sperm on the top, spin it, so the sperm will be filtered to two gradients, and at the bottom, the collection will be giving me good quality sperm. Quantity will be less, but quality of this separation is excellent. So when it is for ICSI, in case of OAT samples, this is the ideal method. It is akin to a water filtration bed in which large sand, small sand, coarse pebbles pour the dirty water, collect the pure water. So it is a gradient separation or systemic separation. Both the methods are excellent, but a skillful semenologist will detect or check which is a good method, which is going to give him a higher yield, and then decide, at the same time keeping into consideration the egg quality of the patient also, how much he is proliferating. So this is the parameter by which we can select whether you do an IVF or an ICSI. Or initially, if the sperm are good, lady is perfectly all right, conditions are normal, age factor fantastic, no need. Either a timed intercourse can also work, or an IUI can also work. So it's a step-by-step graded approach towards the techniques. Now, I always say this, tips and tricks which you have to follow. Please, it is not the textbook says this, so do that. Now, why I always judge the total motile sperm count. Now, I tell you simple thing. Suppose the count is 80 million. We are damn happy. That's what we always say. By count kitna hai? We always ask, oh, 80 million, wow. But then suppose I say motility is just 10%. So how much is the active count or the functional sperm count? 8 million. And from that 8 million, 5% are active forward. So you see how much crucial or drastic is the drop. So this is always very important. Always judge the count in million per ml, count in ejaculate, and the functional count, that is the total motile sperm count. And once you process the sample, you see how much sperm you recover from the aspirate, that is known as the recovery rate. So the biggest and the most important yardstick for a very good embryology laboratory is to check initial counts, 0 to 20, 20 to 40, 40 and above. Survival rate, recovery rate, and then you can judge if the recovery rate falls less than 30% of the original. I'm sorry, something is wrong with your embryology lab. The QA/QC is not proper, the media is not proper, the temperature conditions are not proper, or the skill of the embryologist is at the pits. Because many times we say, the original sample is fantastic, how come the recovery is so drastic? Agree. All sperm don't show a fantastic recovery. There may be some few dark sheep who are just not refusing to swim up. So whenever we used to do this, we used to do a routine semen analysis, we used to do a trial wash, and we used to do a 24-hour survival, as we used to check how many of them are really remaining excellent in post 24 hours. Now, many times we see non-motile sperm in a fresh collection also. Now, what is it? Are they alive and they are not moving, or are they dead and they are not moving? Well, stain them. Every sperm or every living cell has a membrane. What we call it as plasma membrane in case of animal cells and cell membrane in case of plant cells. If there is a crack, fracture, or a tear in the membrane, once they are stained, the stain will permeate it and the sperm will be colored. So this nigrosin eosin test or a plain eosin test will help us detect which ones are alive. They may be non-motile. So remember, non-motile sperm can be alive, but the ones which are stained pink, they are damaged, and so they always will be non-motile. Now these techniques, they look damn simple, but it really needs a skillful hand and lots of practice to evaluate and judge, along with the stains, the concentration of the stain, the preparation of the smear, the exposure time, etc., etc. Well, advanced machines, technology has got no breaks. We can do CASA, just put in a sperm, you can get all the things you want, you can stain them, you get all the things you want. But remember, behind every machine is a knowledgeable human being. Now, last and the most important thing, morphology. Is it informative or just decorative? It's informative. Now, this is an egg. You will feel that the sperm comes and enters. No, the sperm will approach, it will bend at an angle, it will touch. So the pyriform shape, it will tell how much area of the sperm acrosome has touched the ulema. If it is a thin tapering head, just a little part will touch. If it's a globoid sperm, only one dot will touch. When it is a pyriform shape, a little critical area of in contact of the acrosome along with the sperm, along with the ulema, is a very decisive factor which will tell us the shape of the things to come. Now, all will say, oh, fantastic sperm. Now, from this, only one sperm looks near normal. I'm just holding my breath, allowing you to judge which is normal. The only normal is number 4. One has got a vacuole, the acrosome shape is not there properly, and the attachment is not proper. Number 2, shape is brinjal shape. Number 3 has got swollen midpiece. Number 11 has got a fragmented head or multiple vacuoles. Even number 21 has got multiple vacuoles. Believe me, when you see the sperm stained under super high magnifications, in case of OAT sample, I challenge you, you will not get a single sperm which is fantastically ideal. Now, these are all the stain preparations of sperm. And the sad part is, these abnormal sperm also have got fantastic locomotion. So we get puzzled. Only this number 8 is told it is having near normal requisites. The one in number 33 is just a pinhead. It doesn't have the acrosome at all. You see how much ghastly the figure is. But all these are present. Now, unfortunately, there is no remedy. There is no medication. Majority of the times it is psychosomatic stress, because we have got the hypothalamo-pituitary gonadal axis. Now, hypothalamus has got two cells, paraventricular and supraoptic. Now, they have the releasing factors which target the pituitary, anterior hypophysis. Now, the sad part is they are very close to the center of depression. Now, many of the times, patients of infertility are always under some sort of depression, or they are undergoing some treatment. So what happens? One overlaps the other. So the whole axis is shifted, and the Sertoli cell in which these ones are cultivated, see over here, they get abnormal. So this may be a temporary phase. It's like just students when the examination approaches, we get tensed up, we forget what food is, we forget what a movie is. But once the examination is over, we are back to normal, whispering or bubbling with joy. So that means that horrific phase has gone away. It's like a parabola. It has a peak and two small edges. So counseling is very important. Suppose we tell a person, no, sperm report is bad, you have to go in for donor. No, you can't do that. Even a presence of 1 or 2 sperm in the semen sample, you should give him hope. Okay, well, they can shoot up, there can be a temporary suppression, or there can be erratic counts. So Sertoli cell are the architects which holds the key to the morphology of the sperm. Now, still today, there are many unanswered questions. People say, what must be the minimal sperm count beyond which I can't do an IUI? अब तो मैं नहीं कर पाऊंगा ना, अरे मेरे तो खाली 5 million है. You have got a 4 million count. You have got a 3 ml volume. That is 4 3s are 12 million is a total ejaculate. Imagine your motility is 70%. So you are having a healthy 7 to 8 million motile count. And post-wash, even if your count is 3 million or 2 million with good motility, you still have a chance of getting pregnant by IUI. Now there is another test, DNA fragmentation. It has taken up the embryology by storm. Well, it is not at all 100% sure. It is not evidence-based, because what does it do is, these are the number of causes. Okay? And this leads to the DNA damage, whether it's a double strand break or whether it's a single strand break. There are common techniques, comet assay, the sperm chromatin structure, or the sperm chromatin dispersion. Now, this is what the sperm chromatin dispersion test does. The chromatin condenses, there is a presence of halo. If there is an absence of a halo, that means DNA damage is there. Once the membrane is broken and the DNA oozes out, it causes a flower-like effect. But still, well, sorry to say, some people having a very severe DNA fragmentation also have become spontaneously pregnant. Such is the miracle of human pregnancy. What should be done? This is my last slide. Sperm have heads but no brains. You have to use our brains when we use their heads. Okay, thank you very much. Hello. Hello.
Hello.
Yeah, ma'am. I'm done.
Thank you very much, sir. Rafia, over to you for question answer session. Please read out questions for Sir, if any.
Yes, sir. So how can male infertility due to obstruction can be treated?
Obstructive azoospermia is what? It is a blockage in the vas deferens. Now, either the blockage can be rectified, vasa-vasa religation can be done, or if the blockage is severe, then you have to do is percutaneous epididymal sperm injection, aspirate the sperm from the chorda epididymis, and go ahead with an ICSI procedure.
Okay. So then, are there any food products related to abnormal spermatogenesis?
Are there any?
Food products.
Food products?
Yeah.
No, nothing.
Then what is viability?
See, but there are, one second, there are habits. Alcohol, tobacco. tobacco nicotine contamination, it involves into a lazy cilia syndrome. But unfortunately, there are not been many papers which leads to alcohol for the reason of sperm disorders. Unfortunately, no. Yeah, carry on.
Okay, sir. Yeah. What is viability of spermatogenesis suppression therapy and what are the limitations?
See, I tell you, what we are doing in embryology is that we are subjecting our gametes, whether it's a sperm or an egg. Now, the egg is a little difficult because they are not easily available. Okay. Whereas in case of sperm, they are easily available. So there are experiments on them, subjecting them to various degrees of temperature, various methods of separation, post-separation, see the survival rate and the functioning capacity. But I tell you, we have seen that on paper, they really look very lucrative. But actually, the same patients, when treated in the actual treatment, the results are sometimes quite contrary. Now, it doesn't mean that a person whose sperm survival is 80% of the original sample will be able to get a first pregnancy in an IUI. Like similarly, in an IUI also, there are 3 cycles you have to do. 1, 2, 3 back to back. People always say first cycle result nahi aaya, chalo, we are going away. No, the majority of the results come in the second and the third cycle of IUI because the first cycle stimulates your redundant ovaries with the hormones, which become receptive in the next stage. So the second and the third cycle give you the maximum result. So it is a patience game in embryology. It does not mean that it's a long, see, we always convince the patient. You have waited for 7 to 8 years because in India, there are 2 types of patients. One who panic within 6 months of marriage and another who panic after 10 years of marriage. The between time they leave to God. You got it? Or they lose some quacks. And in doing the quackery, they damage the already existing good mechanism of the body. Carry on.
Yeah. So what is the significance of DNA fragmentation test in human sperm? Since at the time of ICSI, the sperm is selected whose DNA integrity is not known.
That's a very good question. Now we are selecting the gametes whom we have not worked on. We are doing the test on the same person's gametes prior to 2 or 3 days. And every sperm ejaculated every day and every egg produced every cycle is different. You got it? So what it happens, what it does is it gives us a generalized opinion. You got my point. Like we say, a team of cricketers who has represented India are supposed to be the best. But they were the best when they were performing well prior. On that day, they might fail. At the same time, there might be another good cohort of people who are there, but who have not been given an opportunity. So it just gives you a generalized viewpoint. कि हाँ भाई उसका semen counts fine. Now today you do a count 6 million, 7 million, 12 million, and suddenly one report says 80 million. Well, there can be cases, but the percentage of such things occurring is less. So we have to read the reports, and these treatment has to go for certain time limit where we can take a judgment over the days to come. One report doesn't make anything. Mark my words.
Yeah.
Next.
Yes, sir. If a person with previous history of testicular trauma, so a person with previous history of testicular trauma can still produce normal sperm?
That depends upon how severe the trauma is. That depends upon whether both the testes are affected or not. Okay. That depends upon whether he is able to produce live sperm in the semen or not. Now, when we do a semen analysis, the presence of sperm doesn't mean. Now, there were tests like zona penetration tests or hamster oocyte penetration tests. But unfortunately, we cannot import hamster oocytes because there is a lot of red tapeism and bureaucracy in the import and export. So what we can do worst, that suppose you have got some extra eggs from a patient with their permission, we try to fertilize those eggs with that husband's sperm and see whether they are capable of fertilizing or not.
Next question is, what are the advantages of yawing movement of sperm, and does it play any role in sperm selection?
Well, good yawing means good kinetometrics. Simple. If a yawing is negative, it is useless. But when we are doing ICSI, unfortunately, we are just catching when I'm throwing him in. So all the kinetometrics go for a toss in ICSI. But still, originally, if a person exhibits any negative symptom, means that gamete is defective.
Okay.
So yawing is a very skillful method. I mean, a skillful observation done by a very skillful semenologist, because all the sperm moves so fast. So, this moment is very necessary. Not like this also, because it's like the screwing action which will denude the zona. That's very important, because when they touch the zona or when they touch the cumulus cells, they liberate hyes, and hyaluronidase dissolves the granulosa cells. Why? Because it breaks the cell-to-cell junctions or the desmosomes, and this clears the pathway. So this becomes absolutely circular. So one lucky sperm does the membrane reaction. If he is normal, then good.
Okay, sir. So, is there any risk associated with electro ejaculation?
No, there is no risk. The surgeon has to be careful, because the voltage is slowly increased from 5 volts to 50 volts, and there is a cutoff. Otherwise, what it will do the tissue scarring by the scalding of the inner sensitive tissues. Effect on the sperm will be none. But as I said, it is documented that the sperm ejaculated after electroejaculation obviously are a little poor in motility, but they have been used for ICSI with success.
So, what should be the minimum time duration from home to lab for semen sample?
Jitna jaldi utna acha. The earlier the better, the faster the better. I always believe that, because see, once a patient collects the traffic trauma, how he comes in, he is under stress, etc., etc., etc. So I would say I would put the barrier at 25 to 30 minutes. The patient will get you in 45 to 50 minutes. Tell him, jitna ho sake utna jaldi. The earlier the better.
Okay, sir. So, are there any genetic or aging issues related to male
Sperm.
infertility?
Well,
Yeah.
some say the gentleman can produce sperm in a functional condition as much as 70 to 75 years. But some studies had found that post 50, the activation potential gradually diminishes. That was the latest trend. But we get the reports, father at age of 70, father at 75, और करिश्मा कुदरत का वैसा होता है.
Yeah. So, what is the reason of low motility of sperm after electroejaculation?
That is not yet documented, unfortunately. There is so much literature, everybody has stated the same thing, that it is still a mystery that post electroejaculation, maybe the modus operandi by which they are brought out, or they are literally being, see, because we are giving a stimulus to the prostate and the seminal vesicle only. We are vibrating them, we are giving them an orgasmic effect. So the nervous control activates the musculature, and the epididymis is also bound by circular and columnar longitudinal muscles. They, by the nervous impacts, they contract and they ooze out the sperm which is collected in them.
Yeah. Thank you so much, sir. Ma'am, please take over the session.
Thank you, sir, for a wonderful presentation. We were all kept unwavering for the entire session. Really an informative one. Thank you, sir.
Okay.
Shweta, please take over the session for vote of thanks.
Shweta, you unmute your mic. Shweta, unmute your mic.
Yes.
That's better.
Honorable Vice Chancellor Dr. Shashank Dalvi sir, Honorable Pro Vice Chancellor Dr. Nitin Kadam sir, respected Director Dr. Mansi Thakur ma'am, respected Coordinator Dr. Minimol ma'am, and our most valued invited speakers and guests and participants, it's my privilege to have been asked to propose vote of thanks on this occasion. So, on the behalf of the Department of Clinical Embryology of MGM School of Biomedical Science, a unit of MGM IHS, I, Shweta Gowda, extend a very hearty vote of thanks to all the speakers for sharing with us their knowledge and opinion today regarding the assisted reproductive technology on this national level webinar. A big thank you to Dr. Karsiavari sir for his efforts to convey us the andrology topic in much depth. I would also like to thank Dr. Pratiksha Joshi ma'am for giving us such a valuable information on ICSI. I would also like to thank Ms. Deol Bose Pandey ma'am for a valuable information on embryo culture environment. Further, I extend my thanks to the organizing committee, organizing coordinator Dr. Mini ma'am, the head of the technical team Mr. Anil Khandare sir for their enormous efforts, my dedicated colleagues of the clinical department of embryology, and last but not the least, our amazing audience. Thank you for your patient hearing and joining us and making this webinar a great success. Thank you all.
Yeah, I would just like to add that anybody who would like any further information, you can just email me. My email is kersiavari@hotmail.com, or you can WhatsApp me 9322405935. Okay. So Dr. Mini, over to you.
Thank you, sir. Thank you very much. Now we'll wind up the session. Now I request all participants to fill in the feedback form mailed to you to procure your certificate shortly. Thank you very much.
Okay, thank you, ma'am. It was a pleasure meeting all of you. And again, I'm thankful to your entire management, entire support staff. Thank you.
Thank you.